Okay, so this is our homepage, as most of you will be pretty familiar with now, for all things bioinformatics. And so far, the people that have joined us for all the sessions, we've gone through intro to R, plotting in R, tidyverse, reproducibility. We've covered a lot of topics and all the way through to Bioconductor. And so in this session, we will refer back to predominantly Bioconductor. If you haven't been to the Bioconductor session, it's okay. If one of these sections doesn't make sense, there should be links to take you back there. And if there isn't a link, because we're not perfect, we haven't linked everything perfectly, then you can just hop back into the Bioconductor section and have a little look around. And I can always point you in the right direction. Okay, but we can just click on this link and this will take us to the section on RNA-seq. Okay, and so we have, it's the same kind of session as before. And we have our checks at the top. So this means the course is compiled recently. This is because it compiled today. I reran it this morning with some just extra little tidbits that I wanted to put in. And you also notice that this OS check, so this is the OS operating system check, is failing. And so this, you can always check why it's failing. And you can click on here. And you can see the reason it's failing is it should work on your Mac and Windows systems, okay, if you have the current release. And so the older releases are not working. And we'll see why in a sec. You can install everything you need. IGV is recommended, but we won't actually go into that too much today. You obviously need R and RStudio. And so the big one to add is Salmon. Okay, so Salmon is something specific that we'll be using in RNA-seq for counting. And I'll explain what that is at the end and how to, at the end of this session, or the middle of the session actually, and how to install it. But basically, we're going to be using a package called Herpa, and that's only available in the current release of R, because we made it and we just made it recently. And so it isn't available for the older releases because it didn't exist. And so that's just to reassure you, this is why that is failing, because we're using this. Okay, and you'll also grab all these packages, and then we can get down to the session like here, and you can just click on the slide, and that'll take you to the slides that we're doing. Okay. Okay, so I'm just going to open up the chat. We have some questions. So we have a question about installing with Salmon or with Herpa. I cannot install, this is from John Nicholas, I cannot install Salmon. I got this message, install Conda tools. Salmon has no matches. There are no packages and versions available. Yes. So this is Windows. I'll cover Salmon later on, but unfortunately, Salmon is not actually supported on Windows. So that section about Salmon, there are a couple programs that are commonly used. They are available on Linux machines, and so you might be able to utilize them through, say, the HPC. But directly on your Windows machine, they won't be available to you. Unfortunately, a lot of programs aren't automatically available on Windows because it's much harder to compile on Windows. And it's a whole separate way of doing things. Okay. But we will go over it, and if you are getting into this stuff, then you can look into either running something Linux friendly on your laptop, or you can start using the HPC. Okay, so RNA-seq. Okay, why are we doing this? And so really it offers a really nice way of being able to measure what's happening to gene expression on a genome-wide level. And this allows you to get an idea of what's happening with both annotated and novel transcripts. And so what commonly people are doing when they're doing this is they will have a wild type and the mutant, or a regular tissue and a cancer tissue, and they want to compare the two and see which genes are changing within their defined groups. And so a key idea to remember when you were thinking about RNA-seq is that unless you have some kind of internal control, like a spike, and it's really hard to get this idea of absolute quantification, because there's so many measurement biases. Even a spike in won't necessarily fix that, because you have to worry about things like GC biases, how mappable is that gene itself. And so most of the time, it's much more appropriate to compare levels between sample A versus sample B, rather than gene A versus gene B. And so this is differential gene expression, and we'll be covering how to do that in this morning session. Something we'll cover in the afternoon, which is less frequently used, but also very important, is changes in specific transcripts between conditions. And so what we mean by this is isoform switching. And so there are methods to be able to detect this in a systematic way that actually use a lot of the same rules we'll learn for this first method, okay? And so we will show you how to do that following these same rules. So then it's like a similar workflow and easy for you to add into your analysis. And so most of what we'll be covering will feature data that's publicly available from ENCODE. And so you can click here to look at ENCODE, but we have some RNA-seq replicate 1 and replicate 2. The FASTQ files are here. And then when we get onto later on, we'll talk about differential splicing and transcript analysis, and we'll be using a data set that you can click here to get. Okay. Obviously, these downloads and everything will take some time, but there's no need to start this right this second, because things like the splicing, this mapping may take some time on your laptops anyway, and it's a good thing to do, say, after the session to help you remember what we've covered. What we have done, though, is we do have intermediate files as we go, and I'll try and point those out where you can grab them. So you can grab some data and start to interact with some of the later sections. Okay, so in these sessions, we're going to try and focus on initially getting this differential gene expression between our T cell groups, and then come up with some summary plots. And then later on, we'll look at this splicing factor. And so in this first session today, what we're going to be covering from 10 till 12. Okay, so it's a relatively long session. So we're going to do a little bit about the QC. We're going to talk about mapping and counting, because they're slightly different things. And then we're going to talk about differential gene expression. So this will cover a lot of what you'll want to do in this first session, but it's quite a lot to cover. But I'll try not to leave anyone behind as we go. Okay. So once you've downloaded your FASTQ file, you can start to interact with it using the short read package. And so this is something we covered in the Bioconductor session about FASTQ files, and the link is here. Okay, and so you'll recognize some of these ideas. Okay, and we'll just be implementing for this new file. So whenever we want to do some QC on a data set, quite often you don't need to use the whole data set. You might only need to use a proportion, just because that will give you a fair idea of what's going on, and that will save you processing time. And so to be able to do that, we can use FASTQ sampler and the yield function. And what this does is it will randomly sample that FASTQ file, and then we can read it in. And so here we're providing the FASTQ file, and we're actually providing just directly the link. So we don't have to download it. Then what we're doing is we're, well, we are downloading it, but we're just downloading it in R. And once it's downloaded, we'll then sample this, okay? And so this is the key point here. When we do FASTQ sampler, we're setting up some parameters where we're saying we want to get 1 million reads from here, okay? And so that's a good representative proportion of your FASTQ file to know what's really going on in your FASTQ file. And so once you have these settings up, this is actually just some parameters surrounding the FASTQ file. You won't actually read in that million reads until you use the yield function. And so when you do all this, you'll then have a million reads saved in your object in R called FASTQ, and it's saved as a short read object. And so this has a million reads, and it has a width of 50 cycles. Okay, so that means by 50 cycles, these are 50 base pairs long, the read length. And so we can do some things with this. We can try and get an idea of what the FASTQ properties are just before we get into the alignment to make sure it looks okay. So we touched on some of this functionality with Tom in the Bioconductor sessions, if you were there for that. Basically, we can use the sread function, and what that will do is it will read in the actual reads from this. Okay, so within this object, there'll be several things saved from the FASTQ file. There'll be the reads themselves, and then there'll be the quality scores. Those are the main things you care about, but there's also things like name information. And so using this the sread function, we can actually extract out the read information from the FASTQ. So this is A's, T's, C's, and G's. And then we can use the alphabet by cycle function. And what this will do is it will extract out how frequently each of these bases are seen at each cycle. Okay, so this is at each base pair. So first base pair, second base pair, third base pair. Okay, and this will allow you to assess, is there any bias that I can see over my FASTQs? So we can then put this into ggplot and have a look. So what we're doing here is we're creating a data frame. Okay, and so it's extracting out A, then C, then G, then T. Oh, wrong way. Okay, so we're getting the rows that are A, the rows that are C, the rows that are G. And then we're creating a data frame from these. We're also labeling it with the cycle number. And then which base it is. Okay. And then you can throw this into ggplot, and then you can visualize it. And so you can always see that there's the beginning of a read and the end of the read are two regions that are quite often lower quality or have some variation or biases associated with them. And so you can see that evident here, at least at the beginning of the read. Okay, but then it stabilizes out, and this looks like some pretty okay sequencing. Likewise, you can check for quality score. And so you do pretty much the same thing, but instead of looking at the sread, we look at quality. Okay, so we're going to the same original FASTQ object, and we're extracting out quality. And this time, instead of doing alphabet by cycle, we're going to just do alphabet score. And so what alphabet score does is it summarizes what is the score over the entire read. Okay, and you can see that here of what this looks like. And then we can draw a histogram. What's the proportion of reads that have good quality scores? Okay, and you can see the majority are up here at the top end. And there's some low quality, and it must extend all the way down here to some of this really low quality stuff. But the vast majority is up here, so that's good. If you have something that isn't so biased to the right, then you'll be worried about the quality of your data. Then the final check is we'll take this quality, these quality scores that we extracted from our short read object, and we'll turn them into a matrix by doing as, and then we define it as a matrix. And then we can just do a box plot of these, okay? And so what this is doing is it's taking our quality scores and then we're getting them translated into the numeric values along the way, and we're converting it into a nice matrix that's easily readable by the box plot function. And so from this, you can then get some information about the variability of the scores at each base. And so this is very similar story to this here. Starts off a little bit wonky, but then it figures itself out. Okay. And so by the time it gets into the majority of the read, it's actually very stable and very high quality the majority of the time. So if you have data that looks like this, you're probably pretty happy. It looks reasonable. There's nothing too concerning. And so you can move on to alignment. Okay. So, yeah, that was always the first step whenever you get a sequencing back. You want to be able to check the quality control before you do anything, because you don't want to get disappointed later on, go off the work of aligning, and then realize that the FASTQ looks like crap. Okay. And so once you're sure about your FASTQ, you can then actually see where do these reads go to in the genome. And so because we're doing RNA sequencing, we want to actually do some alignment, which is splice aware. So this means it will recognize that some of the reads will straddle exons. And so we'll have the reads start in one exon, and then it'll end in a different exon. And so you won't be able to just use, say, subread, which is our go-to aligner, on its basic settings, or you won't be able to just use bowtie on its basic settings. You have to use something that can actually process this, because as you would imagine, it's a lot more complicated if it has to consider a fragmented genome to align to, essentially. And so again, we started to touch a little bit on this in Bioconductor sessions, but we'll cover some extra facets of this that are important for when you're doing it for RNA sequencing. First things first, whenever you want to do alignment, it's always about creating that reference genome. Okay, you want to get the reference genome, and you want to get an index. Now, you can get your reference genome from a lot of different places. We've talked about this a little bit in the past. You can look at our genomic file formats session if you want to see places you can get the FASTA file that contains your genome information. But what we like to do is we like to get it from these BS genome packages. The reason we like doing this is because they're version controlled. And so this means if you say, I got the BS genome mouse UCSC MM10, you can then print out your session information. You'll be able to know which date you got it, which version it is. And then if you want to use that genome later on, it's really easy to rebuild. If you downloaded it from somewhere on a given date, it can be much harder to track down that information. Okay, so for the BS genome object, all we have to do is load it into R using the library function. Then it's accessible to us. And then what we're going to do is create some DNA string sets and export them. So first we'll create these names. So these are all the potential chromosomes in this BS genome object. So we have, so what we're going to do is paste on the string cr onto 1 through to 19 plus X, Y, and M. So that's all our different chromosomes. And this is matching the nomenclature used typically for mouse. Okay, and so this means using this, we'll get CRA1, CRA2, etc., etc. And so these are our names. We'll then do an lapply. So what this will do is it'll iterate through our names, and it will do this function where it will just index out of the BS genome object using one name at a time. Okay, so the way this works, right, is this function will be applied to every element in this list. And so each element in this list will be used to index out from here. Okay, so the first time we go through, it's going to be using CRA1, and it will grab out all the information about chromosome 1 from the BS genome object and return it. Because it's an lapply, this will all be returned as a list. So each element in the list will be the sequence information that we just extracted from the BS genome object. We can then attach this to that object we just created as the names. So that means we now, at this point, we have a list with all the sequence information for each chromosome. And then we need to convert it into something called a DNA string set. So at the moment, prior to this, it's a collection of DNA strings. By specifically saying this is a DNA string set, it gains some new attributes. The one we care about is the fact that once it's not just a collection of strings, but once it's not just a collection of DNA strings or a list of DNA strings, and it's specifically a DNA string set, it's then easy to export. Okay, and so there's a function called writeXStringSet, and so that's why the set is important, because we need a string set to be able to export it properly. And we can export the whole thing as a FASTA. Okay, and so what this will do is it will create a nice FASTA file where each string is exported into this FASTA, and each chromosome is broken down. And so if you're familiar with FASTAs, and if you looked inside them, quite often they'll contain multiple DNA sequences, and they'll be separated by headers, which are associated either with chromosomes or different regions of the genome that you care about or have sequenced. And so this will mean we have a nice FASTA file with each of the chromosomes, and they'll be separated with their headers. And so that means we have a nice FASTA file at this point. So this is which we've called BS genome MM10 main chromosomes. And so that's pretty much what we need to be able to build our index. Okay. We can pass it. I mentioned earlier, subread is our favorite alignment package because it's a very good jack of all trades. It doesn't compromise too heavily on any particular parameter, and so it means it's very robust. it isn't specialized for any specific thing, but it's just very good at most things you try and do it with. Okay, so the specific algorithm, though, we'll be using is subjunct. So subjunct is the splice-aware version of subread. And so before we can actually do anything with this, we actually need to build the index. And so we use the build index function. Okay, and so you can see us using this here. The first argument will be the name we want to give it. And this will be the input faster. Okay, and so this is the key other parameter I want to talk about is building an index is super memory intensive. Tom talked about this before, but the default is set to 8 gigs. And so on our system here, we're also using 8 gigs. And so this is RAM memory. And so for most people's laptops, this will be too much. And so you want to look at your properties of your laptop, see what it is, and don't use all of it at once. Okay. And then you might have to let it be to do this because it can be because it is memory intensive. Your computer will run slow while it does it. But once you've built the index, you only need to build it once, unless you want to change the genome or update it. Okay, so once you have your index, it's a good question from Nathaniel. Is there a rule of thumb for memory available and what fraction to use? I'm not sure if there's a rule of thumb. I specifically, when I've done this locally, is I go for about half. But I don't know if, let's see. JD's here. I don't know if JD can hear me. I don't know if he has any opinions. He can chuck them in the chat. But I typically go for around half because I think that won't overkill your computer. It also depends on what else you want to do. Other times I might go for, yeah, so if you have a 16 gigs of RAM on your laptop, 8 gigs could still be okay. But if you want to be doing other things, you have to just basically remember that half your computer's normal processing is essentially gone. And so it's going to be considerably slower. So it's all contextual to what you're doing. If, for example, you're running it overnight, you can push that up to 9/10 of your RAM or something like that. It all depends on how and when you're running it. Never do all of it though, because your computer always needs some just to survive. Sorry. Okay, so because we're doing RNA-seq, we're specifically going to be doing subjunct, and the reason, as I've mentioned like 3 or 4 times at this point, is because it's splice aware. And so when we say splice aware, it's going to be looking for these exons and intron junctions to be able to map across. However, some of these intron and exon sites don't look normal. Okay, there might be these non-canonical sites, maybe spanning very large distances, or with like irregular organizations. Okay, so in those cases, just running subjunct with the basic settings won't actually pick those irregular isoforms up. And so what we can do is we can actually provide an annotation file as well to help support subjunct in its annotations and its mapping. And so we can provide this as either an SAF format or a GTF format. I think a lot of you will be familiar with the GTF format because these are the ones that whenever you have an annotation file, you can readily download off of places like UCSC. But the SAF is actually a really easy file to generate yourself. And so we'll show you how to make an SAF format that will match your BS genome generated faster. Okay. So for the SAF format, we just need a table with every exon's chromosome location and a ID associated with it. And so to get this information, we can use the TXDB objects. And so the BS genome, if you remember, contains all the sequence information. The TXDB is the transcript database. And so that contains all the transcript information. And they're kept equal, like lockstep with each other, so that TXDB always relates to BS genome. And so to grab out our transcript information, we specifically want to get exons for this. So we use the exons function on the TXDB object. And we're going to need both the transcript ID and the gene ID. Okay. Now this makes sense, right? Because you want to know what the transcript gene is associated with, but you actually want to know also what each transcript is. Okay. And so then we subset this to anything with a single gene ID. Sometimes you get multiple gene IDs associated with a transcript, and that's could be just because the annotation isn't quite right yet. People are still figuring it out. And so we do this to simplify this down. This won't filter out that many genes or transcripts, but it will stop you from having an error later on. So this is an important step. But you can always check what you're filtering out at this stage to make sure it isn't something you're super interested in. And so once you have your exons as a G range, okay, so this is what the product looks like, right? You have every single exon. You have the transcript ID. And there'll also be a gene ID, but it's just not fit on here. Once you have that, you can convert it to a data frame. And inside the final data frame, to make the SAF format, okay, we had Nathaniel had a question just to clarify what we filtered out. Okay, so as I was saying, this is asking what the length of the gene ID column is. And so sometimes you can have multiple gene IDs per transcript just because the annotation is still maturing. And so this is just ensuring you have just the single annotation. Okay. And so this is really important later on. So this removes anything with multiple annotations. Okay. So it's imperfect, but it's for the majority of what you care about, it will keep it in. This removes only a very small fraction of the reads or of the genes. But as I said before, if when you run this for the first time, you should have a look, okay? Because there's a few genes, but maybe they're ones you care about. But most of the time, you will not care about every single specific gene, okay? And there isn't necessarily a robust way to deal with this without manual curation. So the way we deal with it is we just remove all, and we'll worry about if it's a specific gene we care about later on. Okay. So now you have all this information, you want to turn it into a data frame, which is in the appropriate format for subread to understand it. And so it's expecting a gene ID in the first column, chromosome in the second column, the start in the third column, the end in the fourth column, and the strand information in the fifth column. Once you have all this, that is your SAF format. Okay? And the nice thing about this is you don't even have to export it. Okay, so you can save it as an SAF, and then you'll be able to go straight into mapping. Okay? And so we'll run the subjunct function. We'll use the indexing here. So this is our index. This is our FASTQ file. We want a BAM file as our output. We want this BAM file to be called treg1.bam. And then, so this is related to the SAF format. Okay, so we're saying we want to use an annotation to make this, the read assignment better. The annotation.ext, this is what where we're getting the annotation. So we're getting it from the SAF object, which we just created. Then it will ask you the question, is it a GTF? No. And so if it's not a GTF, it'll assume it's an SAF. Okay. And then lastly, it asks for the number of threads. Okay. And so number of threads is how many different threads will be run through for the mapping. Again, this will use up your computer's processing. So you just have to be wary of how many you use of this, but 4 should be fine for everyone. Okay, so obviously the indexing and this step may take a little while. Indexing probably longer, depending on your genome size. And then this, subjunct step is where you actually produce your BAM file. Once you've done that, and it's gone through, it's taken its time, you can then have to do a couple of steps before you be able to use this in IGV and for any other downstream analysis in R, and that is sorting and indexing. Okay, and these two steps really just make the BAM file a lot easier computationally for the file to be used. And so this uses the package RSAM tools, and it will have sort BAM and index BAM. And so the sort BAM will just basically, it's going to organize it by position in the genome. And so what you give as your input is the BAM file, and then you give the name. The confusing thing about this function is it's going to add .bam to whatever name you give it. So even though you have to specify that this is the input file with .bam, it's going to add .bam onto here, whatever you put. So if you put sorted treg1.bam, it will become sorted treg1.bam.bam. It's kind of silly because it's not intuitive, but hey, that's just a tidbit to remember. And then we have indexing here. So that's very straightforward. You'll just give it the BAM file that you want to index. So this is our product of the sorting, our sorted treg.bam. And once you've done that, you can then open it in IGV. You can have a look at it, see where all the reads are. Okay, and so this is an example screenshot of what that might look like. It'll show every read, and it shows where the introns are as well. So it will show the spanning junctions between exons, or between where the read started, where it broke, and then where it started again. And so that's what these kind of smaller gray lines will be. But that'll be clearer once you drag the data in and start to play with yourself. Okay. What we won't be covering is converting that to a bigwig, I don't think. But we do cover that in the bioconductor sections in the genomic scores session, I believe. So let me just. So in Introduction to Bioconductor and genomic scores, yeah. So in this section, it will cover taking something that is a BAM file and converting it into something that is a bigwig. So if that's one of your aims, to get tracks over the entire region, then you can do it in there. Okay, but we won't go into details of that because we are not adding anything to that workflow here. Okay, so counting. So once you've done your mapping, you know where every single read goes, but really you want to have a specific count for each gene as opposed to just knowing where the read goes. That's where we're aiming for. And so to do that, we need to assign each read to a gene. But before we can do that, again, we need to extract out all this gene model information so we can use it for the counting. Okay, so this is a very similar step to what we did to build the SAF. We're going to use our TXDB object again. But this time, instead of using the exons function, we're going to use the exons by function. And we get exons by gene. Okay, and so what we get as a product of this is something called a compressed G-ranges list. Now, what does that mean? It's going to create, it's basically a list of G-ranges. You can infer that from the name, but contained within that is exons, because we asked for exons by, and it's exons for every single gene. Okay, and so that's what each element in the list contains. So this first element in the list, you can see it corresponds to this gene. And this is the entree gene ID. You can see the next one is starting down here, and it'll have every single gene in the TXDB object. And then every entry within the G range that's contained within this entry corresponds to an exon. Okay. And all of these exons have IDs associated with them. Okay. So at this point, you know every position in the genome that is annotated that you care about for RNA sequencing. Okay. And so at this point, we can now use a function which we've met before called summarize overlaps to actually infer how many of your reads overlap with each exon. And so to do this, we use the BAM file function. We specify what BAM file we care about. And importantly, the yield size. Okay. And so what this is doing is it's telling, again, this is similar to the FastQ sampler function that we covered at the beginning. This is setting up some parameters. It's not loading in the BAM. It's setting up some parameters saying, hey, I have this BAM file that's here. And when we look at this BAM file, I want to consider 10,000 reads at once. No more than 10,000. We can just do 10,000 at once and that's it. And then when we do summarize overlaps, and we're specifically saying we want to use my BAM, okay, what it's going to do is it's going to take all the gene exons we give it, look at my BAM, and it's going to count how many reads in my BAM overlap with these exons. And because we've limited the yield size, it's only going to consider 10,000. And so this really, again, it helps control your memory that you use. And this makes it doable to do it on your laptop if you dial this down. And so when you try this out yourself, if you do this and your laptop freezes or dies, then you can just reduce the number, okay? If you're doing it and you're doing like 1,000 and your laptop has no issues, but it's taking about 2 weeks to do a single count, then maybe you can scale the number up, okay? I can't really give you a specific guidelines or number for this because it is very variable depending on each laptop. But I'd say start with 10,000 and see how you go, okay? This is a lot of intuition as you get used to what data your specific laptop can handle. Okay. And so at this point, once you've done summarize overlaps, it produces this object called a range summarized experiment. Okay, and this is one of these bioconductor objects that we'll keep meeting and can be quite confusing for people. We're going to meet some more of similar objects later on. I think the confusing thing about it is it's not flat. It's not just a nice rectangular table. It contains multitudes within it, okay? And whenever you type in the name of it, you'll get a response and it'll tell you some things about it, but you don't really know what's inside. And so a good way to think about these objects, often they're specialist objects that correspond to a specific package, but range summarized experiment is one that's core to Bioconductor, so it will reappear. And so the way to think about these objects is they're a container, they contain lots of things, they're almost like a list, but what's contained within them is just obscured a little bit. And it's obscured with a reason. As you get more used to working with these, it actually becomes a lot easier because it simplifies things. And what they do is they have simple functions that you can use to grab specific parts out. So there's specific ways to grab out the names associated, or the ranges, or this count information, okay? And we'll get more familiar with this as we go along. Okay. And so one of the big things we're going to show you that's specific to RNA-seq here is often when you actually look at, say, we consider counting in gene models, when you look at the exons themselves, and maybe they'll overlap. And so whenever you want to count by exon, say you want to look at transcript information, and we want to compare this transcript to this transcript to this transcript, there's actually an overlap in this first section, which makes it really hard to differentiate between transcript 1, 2, and 3. Because then which transcript do you associate each read with? And so the way we deal with this is we create something called a disjoint exon. Okay, and so this is a really important step if you want to ever deal with isoforms or different transcripts for a gene. And so the idea is you build up a model that's based on individual exons as opposed to individual genes, and then you do the counting that way. And so what we first do is we create disjoint exons. So we don't actually care about this exon versus this exon by this exon. We care about these individual regions that are unique. Okay. And so you can see here, this is our disjoint exon build. And so we have this region that's found in all 3 transcripts. We have this region that's found in just 2, and then we have this region which is found in 1. And by using the combination of the 3 and the counts across them, when we do our differential transcript analysis later, we can infer which is which. And so here we are using the disjoint exons function. And so this is similar to the exons by or the exons function we use later on. It's part of genomic features. And we just give it a TXDB object. And then what we're going to do here is we also are going to create our own name. Okay, to associate with this. So we're going to take the gene ID and exonic part that's produced from this. We're going to add them together. And we're going to use that to name our object. Okay. And so you can see we have exonic part and gene ID. So that's going to be the names here. So we have the gene and exonic part 1, gene exonic part 2. This is just a self-driven name to add to these disjointed exons. But what we're producing is essentially a GRanges object, just like we have done many, many times before, where we have the range, we have some gene ID, we have some transcript information, and yeah, we'll also have some exon ID as well. But that's all hidden. And so you can use, once you've built a disjoint exons annotation, you can summarize overlaps in exactly the same way over it to get counts in your disjoint exons. And so a key thing here, which is a new parameter, is you want to set inter.feature to false. And so this allows you to it allows counts to be more fluid and count over multiple regions. Okay. So I talked a little bit about range summarized experiments earlier. And now we have 2 of them. We have one that has our disjoint exons, and we have one that has our gene counts. Okay. So you can see them both here, our gene counts and our exon counts. And to access the information within the range summarized experiment, one of the most important functions you'll use is the assay function. What the assay function does is it basically extracts counts. When you have these experiment objects, you can grab information from inside them using different accessor functions depending on what you care about. But the assay function always returns basically the experimental value and the count information associated with it. Okay. And so in this case, this is going to be how many reads were assigned to each of these different regions. And so you can see what this looks like. If we look at the first 2 rows of each, we look at the exon one, it will have the name of the exon. Okay, so this, if you remember, we created this, it's the gene ID and the ID of the specific disjoint exon. And we have a count of 0. We go to the next exon ID and we have a count of 1. Well, the gene level counts will just have each gene. So you can see the first gene, second gene. And you can see actually that this count sums up to here. So there's going to be no other counts within this gene. Okay, so we have the same information going into here. It's just being counted in slightly different ways. That's the point that we want to make. Okay, so at this point we've done our alignment, we've done our counting. Okay, we are ready to do differential gene analysis. However, there is an alternative way to get to this same point, and this alternative can be a lot faster and easier. depending on your like processing and things like that. And it can often give you an equivalent answer. And so that's using an approach of transcript quantification, direct transcript quantification, okay? And so in this, what we just did was this two-step approach where we do alignment, then we do counting. There's been recently, or over the past decade, much more leaning towards using a k-mer counting approach towards quantifying your transcripts. And so some of you may have heard of packages like Salmon and Callisto, amongst many others. And the way they work is, instead of trying to map each FASTQ file that you get or FASTQ read that you get and directly detailing this has to be in chromosome 3 at position 1 megabase and 270 kilobases or wherever, and directly annotating where each read is, it will look at the FASTQ file and the read, and it will look for sequences within it that seem to be unique or rare within the genome. And it will look at several of these sequences, and if several of them are associated with a specific gene, then it will give that gene a count. Okay, at that point, it's not really considering where that read belongs specifically on the genome. It's just looking at the most pertinent information within that read and using that to infer which gene it came from. Okay, I know that might seem a little confusing, but it is, like, the outcome of this is it's a lot faster because it doesn't need to know exactly where the read belonged. It's just doing some counting instead. And so this whole step skips alignment and goes straight to counting. So this can be really good, really efficient and quick, and it largely has the same results. The big thing you won't get from this is you won't have an aligned file that you'll be able to look at on IGV and produce tracks and things like that. And so it is a nice alternative, but you do lose something along the way, but you do gain speed. So for those of you working on your laptops, this might be a nice alternative. And so the one we're going to talk about is Salmon. As we mentioned at the beginning, with tools that have become more and more specialist, there's less and less support for machines that aren't Mac or Linux. And so this actually isn't possible on Windows. So apologies to those who are using a Windows machine. Again, what I said at the beginning is there are ways to be able to run Linux on your Windows. And also there are ways to sign up and use the HPC, the high performance computer. And you'd be able to do this kind of stuff on there. So this code is still useful. It's just maybe not useful right now, right this second to you. Okay, so apologies. The other thing about this is there's no actual package in R for Salmon. Okay, you can, like I said, you can install it on Mac and Linux using the Anaconda package repository. And so if we click here, if you ever want to see if something is Windows specific or whether it works on Windows or whether it works on Linux or OS, you can come to somewhere like Anaconda. Most biological tools are contained within here, within specifically the Bioconda section. And you can see here it's Linux or OS, okay? And so this repository is similar to, say, Bioconductor. It's a place where people put their packages, they're version controlled, they should work. It's less rigorous than Bioconductor. It's not as tested as much, because pretty much you can just upload something to Bioconductor almost, a Bioconda even. But it still has a history associated with it and does have some checks. So it is a good place to get software. And the issue with it is it's a separate system of managing things outside of R. So if you're learning R and building up these skills for the first time, it's a whole other thing to learn. And so what we did recently is we created a package called Herper, which allows you to interact with Conda from within R. And so the way Conda works is it's basically a package management system, and it creates these environments which you can then use at any time. So you create an environment for RNAseq, and it will install the software associated with RNAseq that you want in this environment. And then you can activate this environment and all this software will be available to you. And so what we do with Herper is we take advantage of this and then just interact with Conda through R. So you never have to leave R, but you can use all these other programs that just aren't in R. And so to start off with, what we can do is we can install Herper. It's a Bioconductor product. So you can just use BioC Manager and then you load it in. And then the installation is very straightforward. You can use the function install Conda tools. Okay, so in this, we're going to give it the information. We want to install Salmon, and we want to install it into an environment called RNA-seq analysis. And so what Herper does behind the scenes when it's doing this is it installs Conda on your machine, and it installs the most minimal version of Conda, and that's called mini Conda. And then within mini Conda, it creates a new environment into which Salmon will be installed. And so this environment is called RNA-seq analysis. And so if you're joining us for ChIP-seq tomorrow, you'll be able to use exactly the same mini Conda, and you'll be able to create a new environment, which you can say ChIP-seq analysis, and you can install Max in there for peak calling. There's some additional arguments for this. So you can specify where mini Conda is installed. As I was alluding to there, you can use mini Conda, like the core part of Conda, mini Conda. You can use that again and again and just have lots of environments. And these are little partitions where you save whichever programs that you want to use for that specific analysis. And so you can keep your path to mini Conda consistent. There is a default, though, that's associated with a package called reticulate, which is the way that Python is handled within R. And so if you want to just leave it blank, you can just leave it blank and it'll just keep installing to the same place. It depends if you are a Conda person and you've used Conda before, you can get that control if you want. The other important argument, I think, is update env. And so say we want to install something else in here. Say I want to do Salmon and I want to do Callisto. You can add update env onto this, and you say update env equals true. And then subsequently, you can also install Callisto. And what that will do is it will update this environment to also have Callisto in it. If you don't do that, it will tell you that environment already exists and you can't install something, create a new environment. Okay. So that is the installation of Salmon. It should be fairly straightforward. And because it's the package that we maintain, if you have any issues, you just reach out to us and we'll fix it as quick as possible. Okay. But the whole idea is with Herper is you can get access to a whole ton of programs which aren't available typically in R. There's a lot in Conda beyond just RNA-seq stuff. So anytime you find some software that isn't R specific and you want to use it, you have the R skills, you can use maybe Conda with Herper to be able to use them and then just stay within a situation that you're familiar with. Once you have Salmon installed, you have to build an index. Okay, so just like we did with subread, you have to build an index. And so you can extract, to do that, you need specifically the transcript sequences. Okay, and so there's a function we haven't touched on, extract transcript seqs. And this will give you all your transcript information from the TXDB and the BS genome, okay? So the way this is working is it's looking in BS genome and it's using the TXDB object to figure out which sequences to extract out of BS genome. So it's slightly different to subread, right, where you give it the FASTA for everything. Then you tell it where the annotation is, and it figures it out. This, we just care about the FASTA information for the transcripts, because we don't care about anywhere else in the genome. And then once you've done that, you have a string set, so you can just export this with the right X string set. You can also export some decoy sequences, okay? And so what decoy sequences do is they allow Salmon to consider regions outside of the regions you care about, so regions that aren't transcripts, when mapping. And so that means basically, if you have a region of the transcriptome that's very unique, very interesting looking, but actually is very common within the non-coding part of the genome, it might downweight some of that a little bit when it's assigning a count to that region, because maybe it's just DNA contamination. And so a decoy can actually increase your mapping accuracy. That said, and it's generally recommended. That said, it does actually, as you would imagine, it's an extra step, so it does make things a little bit slower. And so depending on the way you're using this, like the way we use Salmon and alignment is we have our Salmon, and this is our quick, easy pass. We can look at the count, see if things make sense. And then if we're producing a final experiment, we really care about knowing exactly where things look, then we can actually do a full alignment. And so depending on that cost benefit, maybe a decoy is really important to run, or maybe it's not. Okay, and so with the decoy, you want to be able to extract out the whole genome, just like we did before. This is what we did for subread, right? We create a string set, and so now we have our main chromosome set, and we also have our transcript set. And so together, we save this object. We have all the transcripts and we have all the chromosomes, and we write this out. And so this is going to be our decoy information. Okay, so we have the whole genome. We have both the transcripts and we have the genome sequence itself, and then we need to tell it which parts of the decoy and which parts aren't. And so in this case, we just give it the information of what the main chromosomes are. Okay, and we're writing that to a text file called decoy.txt. And so if you remember, when we exported this, we gave these main chromosomes as names in this list. And so each of these sequences in this FASTA that we just created that has the main chromosome names, these will be directly associated with those sequences. However, the transcript sequences will have different names. They'll be associated with the transcripts. And so they'll still be differentiated as the main chromosomes are the decoy, but the main transcripts are not the decoy and what we're actually caring about aligning to or counting in. Okay, so once you've built up your index, be that a decoy or not decoy index, you then need to run Salmon and do the counting. Oh, no, do the indexing even. We've got the raw materials to do our indexing. Now we actually need to do the indexing. Sorry, I jumped ahead. And so we can still use Herper to do this. And we do this by creating a command to Salmon that we would typically give to our OS. So we'd write this into terminal, or we'd write this on the command prompt, not in R. Okay, and this is the command we typically do. We do Salmon, we say we want to do index. We have this as our input, and this is our faster. Okay, so that's our index and the faster. Okay, and so as I said before, Herper allows you to run things within R, and so we use the function with conda-env to do this. And so what we're going to do is we're going to recreate this command and run it from within R. And so we do, whenever we want to create an OS command within R, we use system or system2 to create a system call. And what with conda-env is doing is making Salmon available to us. Okay, so we're saying with conda-env, I care about the programs or the environment or the programs within the environment RNA-seq analysis. So that means Salmon becomes available to us. And then we pass this command, system2, the command is Salmon. And the arguments we give to Salmon are index minus I. We have the index name. So we save that up here. And the faster file, faster.tx here. And then we add some extra system information here. So standard out just means if there's any errors when it does the system command. Okay, so this is all one function essentially. It will report them out for you so you can see them. You can turn this to false and then you won't see any errors or any outputs, but it's good to see them. So we do standard out. Okay, and so with this, it will run a Salmon command, this Salmon command in R without you having to leave R. And so you can run a very similar command if you want to do it with decoys. You just have to add an extra parameter, which is -d, and then you add your decoy.txt information. Okay. So hopefully that doesn't, it's several steps and it's a little bit complicated how to run these non-R programs if you're doing it for the first time. But I just want to iterate that this process should make life a lot easier for you than having to do things separately. The other nice thing about it is now that you've done this, you can then, if you're doing this, say in an R Markdown, there is a function to list the programs and packages that are in this Conda environment. So you'll be able to get the versions of everything. And so then you can stay very reproducible. You can also export it as a export all the versions to what's called a YAML file, which you can then save, and then you'll have all the versions and everything associated with this project saved. So it'll be super easy to reproduce. Okay, and so now you've done the index, you have to do the quantification. Okay, so it's just like mapping where you have the indexing, then the alignment step. The big thing about this, obviously, right, is it's skipping alignment and it's going straight to counting. And so, like before, we have this command. Okay, and this is what you'd write if you were writing it directly onto the operating system via terminal or the command prompt in Windows. But in R, we can use this code. Okay, so we know where the FASTQ file is, we know where we want to save it, and we do with conda-env. We get the RNA-seq analysis. We're going to use the RNA-seq analysis environment. We're going to say, basically run this command up here. We're going to do Salmon. We want to do quant. We have these index information. It's going to detect what kind of library you're doing. and the R is what the FASTQ file is. Okay. Okay, so Nathaniel has this question: how do we know the gene without aligning, or we don't know this way? So we do know the gene. Okay, so the way this works, and sorry if it wasn't clear earlier. So instead of knowing, instead of positioning each read in a specific gene, it will look at the read and it will find different sequences which look unique within that read or rare, and it will then compare them to the index. And what the index does is it looks over the entire genome, and for every single gene or every single transcript, it will look again at similar things, see which small sequences are rare. Okay, and then it can compare them between the two. And so this is what they call like informative k-mers, just informative short sequences of like somewhere like 4, 5, 6, 7, 8 long sequences. And so this way, it's not considering the entire 50 base pairs, 75 base pairs, or 100 base pairs. It will find the 3 or 4 that are of these short sequences that are informative. And then it will figure out which gene has these. Okay, so it will be telling you which gene each count or each read belongs to, but it's not going to be placing, say, the start of this read is in this specific position in this gene. It will just know that this read most likely belongs to this gene based on similarity of just the informative parts. Okay. Cool. I hope that makes sense for everyone else as well as Nathaniel now. Okay. And so once you've created this quant.sf file, that's the product. It's called a quant.sf. It's basically a data frame. And there's a package called tximport, which is really good for importing these. We'll cover that in a little bit. But you can actually just use standard libraries to install it. So we can use read.dlim. And what's contained within it is you'll have the length of the transcript, some transcript annotation, and then some information about the number of reads assigned to it and the TPM. Okay, so you can see how it skipped a lot of steps there, but it processes really quickly, which you haven't had a chance to experience, but there is a substantial difference, especially if you're running locally. And like I said, the results are going to be pretty similar. So if you're not interested in just chucking things into IGV straight away, this is a really good alternative. Okay, so there's some exercises and stuff, but I'm hoping to cover cover differential expression now as well. Although I've been talking too much, so we'll see how this goes. But what we'll do is actually we'll do stop for just a couple minutes so that everyone can recover from what we've covered so far. I'm going to grab some water, and then in a couple minutes after this, I'm going to launch a poll as well. Lots of things going on at once. I'm going to launch this poll. You guys can tell me which of these you think is true from what we've covered so far today. We'll take 1 or 2 minute timeout, and then we'll get back into doing differential gene expression, and we'll see how far we can get in the remaining 30 to 40 minutes. Okay. Okay, let's quickly go over this. I'm going to share the results. Okay, I think most people get the idea of salmon. It seems to be the most correct answer overall. Salmon is very fast, but it's not a true alignment. Okay, so there's, like with every tool, pretty much, there's pros and cons. I encourage you to try it out and try out subread and then see what you like. Because as you'll see in this session, there is quite a lot of overlap in the results. once you build an index, it will work for all aligners. Well, this one's a little tricky. So once you build an index, it works for all alignments. In the case of every time you run it with that aligner and on that genome, you can keep using it again and again, okay? But if, say, you do something in subread with the MM10 genome, and then you want to do something in star with the HG19 genome, you will have to build a new index, okay? Even if you want to do something in star with the MM10, so the same genome, you will have to build a new index. It's just each algorithm has an index which is optimized to help that algorithm work as efficiently as possible. As I clarified, disjoint exons help differentiate between different exons. That is correct. If you remember, the disjoint exons basically show each kind of unique pattern of exon presence within transcripts. So in the example we showed, it showed one disjoint part of an exon was present in one transcript, the other part was present in 2 transcripts, and the final part was present in 3 transcripts. They were all part of one exon, but when you did disjoint exons, they were split into 3. And then finally, the SAF file format contains aligned reads. That is false. The SAF file format was an annotation file, which contains just positions of genes. Okay, and we use that to help infer where exons and introns would be, so that if there's non-canonical splicing happening, if a read is over that non-canonical splicing site, it can then be mapped effectively. Because whenever it comes to non-canonical splicing, alignments will do their best, but they often will miss it. Okay, so we'll stop sharing that. Okay, and we'll get into differentials. As I said, we are running a little bit behind, so I think there is a good point in this where maybe we'll get up to that, and we'll maybe have to save some of this section for the next session. But we'll see how we go. I know this session's a particularly long one and is very dense because it contains a lot of the information that people care most about. So we'll just do our best. And as always, keep asking those questions. And I hopefully will be able to answer them for you. Because the last thing I want to do is leave you behind with this. Okay. So there's a lot of stuff in the beginning, which is the same as we covered last time. So I'm just going to skip it real quick. It's basically, what data sets are we working with? Okay. So to start off, we're going to basically consider a couple things to supplement what we just went over, but that will actually increase your workflow, make you work more efficiently when you start to actually do this on a level where you're using an actual experiment. And so what I mean by this is when you do an actual experiment, you'll consider multiple FASTQs at once. And so when we created our range summarized experiment using our summarized counts, we did it with a single file, but you can actually do this with multiple files at once. And so to do that, instead of using the BAM file function, which set up some settings for how you interacted with the BAM file. If you remember, you controlled how many reads you can read in at once. We'll instead use a function called BAM file list. And so what BAM file list is expecting is a named vector of BAM files. So in this case, we have 5 BAM files. We have 2 of one condition and 3 of another condition. Okay. And so we have sorted treg1 or sorted treg act. Okay, sorted treg or sorted treg act. So those are the 2 conditions, and we have 2 replicates and then 3 replicates. And so we've saved this as a vector. We've then named this vector. Okay. So we just use the same names. We just drop the .bam. And then we do exactly the same thing. So instead of just giving this the BAM file name, we give it this vector, and then we use the yield size again. So this means only 10,000 reads in total will be read in at once. And we can then just do summarize overlaps. Okay, so we give it exons in exactly the same way into here. And we give it this basically a BAM config object, which we just created with the BAM file list. And what you'll notice is now this has the same amount of counts or rows, but now we have 5 columns. Okay, so you can see that in the dimensions. And we have the call names here, which were taken from the names. And so doing it this way, you get to work through things much, much quicker because you're addressing several things at once. Obviously, it will be more intense, but that's not more intense for your computer. You're controlling that also with the yield size parameter here. Something to consider, which we'll just touch upon, but this is really just scratching the surface. There is a package called Bioconda Parallel, and this is a way to start parallelizing your workflows, your functions, so that when you start working with these multiple files, they get split up. And so that means you can, instead of it, when you're working on your computer, instead of it considering file 1, then it will consider file 2, then it will consider file 3. If you parallelize, what it will do is it will consider all the files at once. Again, you need a machine to be able to handle this. But if you do have this setup, it will speed things up dramatically. And so this is ideal if you start to work with the HPC. And so it all works through this package called Bioconda Parallel. You can load it in with just library. Obviously, you want to install it first with Bioconda Manager. And then you can set up how many cores you want to work with at once. So in this case, it's all you have to do is set up some parameters, and then you have to register those parameters to be available. Okay, so in this case, we say multi-core param. We want to use 2 cores. And so that means it will run 2 sessions in parallel for you. And so that means on average, it will get through a lot quicker because it's doing things in parallel. You can try and do this on your local computer, but it will run through, it depends on how many cores you have on your computer. And obviously it will start eating up your processor speed. Okay, so that is something to consider, as well as just your memory, but it will be considering, or you use up your processor as well. And you can control it through here. So we have this multi-core param function. And so you can do param multi. And then if you want to do this setup, you just do register function on param multi. Once you've done this, you can also convert it back using the serial param function. What serial param does is it goes back to, instead of considering things in parallel, it considers them back in serial, so one after the other. So this is some more advanced level workflow management stuff, but it's hopefully this quickly touches on it and makes you aware. And you can start to play around with it if you do want to speed up your analysis, especially if you start to work on the HPC. Okay. Yeah. So Nathaniel has a question. When we're limiting the yield, does it just know to go back through the rest after? It doesn't only take 10,000 reads randomly, right? Yes. So you are correct. You are correct. It will go through, because of the style, what you're doing with it. With this, you want to be able to iterate through every single thing. And so it knows that you want to do that. And it will read through the BAM file in a sequential manner, starting at the top. It will read in 10,000. Once it's finished with the first read, it will read in the next one. But it's handling many, many reads at once. And so it is different to the function we covered at the beginning, FastQ sampler, which samples to 1 million reads at once. There is a similar one for FastQ called FastQ Streamer, which works in the same way as this BAM file does, in which it will run through the entire FastQ. And that one is really useful if you ever want to do some trimming or you want to remove reads that are low quality. Okay, cool. But yeah, really key point, if you're doing counting, you want to be able to count every single read, so it takes care of that. Okay, so we're now on to, we've done our counting with summarized overlaps, and we have this range summarized experiment, and we can access it with assay just like before, the assay function, and that'll give us our counts. And as you can see here, we're just going to look at the first 2 rows. It has the first 2 genes, and you can see it's basically a data frame or a matrix, and it has the number of counts per gene per sample. Okay, so at this point, you're in really good shape, right? You have all the count information, and you're ready to do differential gene expression. The nice thing about summarized overlaps, remember this is kind of this abstract object that we have in R, you also contains lots of other information. So we can grab out the row ranges from it. And so this actually tells us, when we consider this gene or this gene, what ranges are associated with that gene? How did we build these counts? And so you can just do row ranges on this summarized experiment, and you can get the ranges back out. Okay, so this means you could save this summarized experiment, the gene counts, and it will have all this extra information associated with it, and it will persevere. So it's one of the strengths of this, you doing things this way. Okay, so differential gene expression with RNA-seq. There's, as you imagine, there's a ton of different ways of doing this. But basically, the key steps are we need to do some normalization. We need to transform the counts potentially, and then we need to calculate the significant difference. And so there's kind of 2 schools of thought for this. One is you start to do some transformation, then you do some normalization, and then you do a kind of standard test over something that's a normal distribution to find your differential expression. The alternative is you normalize the data, but you don't do some transformation. You keep your counts. And you use a test that's actually suitable for counts. And so with most of these kind of normalization approaches and what we're doing in general, you're assuming that the total amount of RNA across your samples is relatively similar. Okay, so this is something that people can run aground of. Say if you're influencing core processes and majority of genes are changing because of you're affecting, say, pol 2 or something like that, that can make your transcript quantification quite hard to understand, because maybe the relative levels between genes are not changing that much, but the total levels are. But most of the time you're interested in a small couple of genes that are changing because of as a direct consequence of some perturbation or some mutant. And so then it becomes much, this is where differential gene expression is strongest. And so the way we handle this is, even though you might have slight fluctuations in your library sizes, so one FastQ versus the other, we normalize those out by considering how many total reads do you have. And so this is how you generate things like simple measures. You may have heard of reads per million or counts per million, and you use these simple scaling factors to do that. these can then be transformed into things like RPKM and TPM, which also consider the gene size. These are great, but they are using less of the data and can fall short of really leveraging data to its full extent. And you're also transforming the data more because you're doing all these calculations, right? And so more sophisticated methods like DESeq rely purely on the raw data, and they consider what the distributions of the data is, both within a single gene and across your whole data set, to infer a lot more about your RNA-seq data, which then allows you to make stronger and more concrete observations about changes in your data set. And so we'll show you the various steps of how things like DESeq work. And so one of the big things it will work on is modeling the relationship between your average expression and the variance. Okay, so you can see here we have on the bottom we have some gene expression, and on the x-axis we have gene expression, on the y-axis we have the variance. Okay, we have some square root of standard deviation. And so you can see there's quite a lot of variance, and that variance changes depending on how much the gene is expressed. This makes sense, right? If you only have a few counts, then you're expecting a lot of variability relative to the few counts, because the relationship for gene expression is a little bit more stochastic. And so by modeling this change along here, you're able to shrink the variance for individual genes. If you consider the variance across the whole data set and create a model for that, then on an individual gene level, you can shrink that variance. And when you shrink variance, you're able to actually increase the ability to call significant differences between genes because the variability is smaller, right? And so there are lots of different ways of doing this, as I mentioned, and we touched on DESeq too, and that's what we're actually going to predominantly focus on. But there's other ones such as EdgeR, VUM, and Limma. Both EdgeR and DESeq2 work on the count data raw. So what we have already is ready for DESeq2. VUM, on the other hand, will actually transform using log2 values. And it's associated to kind of generated this mean dependent weighting, which it can then use with Limma to do differential expression. Okay, and so these are, but all of them have, all three methodologies are really, really popular, and none of them is like, we wouldn't say any of them are particularly worse than the other ones. We just have a favorite in DESeq2. It works very well in Bioconductor, and it fits with a lot of other packages, and that's why we like it. Okay, so how do you set yourself up for DESeq2? I think it's relatively straightforward getting this differential gene expression going. One of the first things you need is essentially some parameters, right? You want to be able to tell DESeq2, what do I care about? Which is sample A? Which is sample B? And so this kind of, the grouping information, you have to create as a data frame here, and we're going to call this data frame metadata. And so what we have here is we're going to have our we have, if you remember, 5 samples. 2 of them are naive cells, and 3 of them are activated cells. It's a T cell data set, so it's all going to be immunology related. And so we can use this as our grouping information. We have 2 replicates of this naive, 3 of the active. Okay. And so what we're doing here is we create a data frame, and each column of the data frame is basically going to be some kind of group information. We only have one group we care about, but you can have multiple groups if you want. And then the row names will be basically whatever column names you had in your gene count object. So we have sorted Treg 1, 2, and 3. And so this means DESeq is going to know that the sample that is sorted Treg 1 is part of the group naive. We then use the function DESeq dataset from matrix function, and this creates your DESeq2 object. So as you can probably infer from the name, we need to provide it with a matrix. Okay, it's going to create this DESeq dataset, but we need to give it a matrix. And so as you saw earlier, if you use the assay function, you can get a matrix from your range summarized experiment. So we just do assay, and we get our gene counts. We can also use row ranges, and this gets our G ranges as well. This is nice because then the ranges are associated with your DESeq object as well. And this is optional, by the way, but it's a nice addition because then the row ranges stay associated, and that will keep persisting with the objects you create. Okay, so in the first argument, we have our count matrix. Second argument is col data, so that's our metadata we created. We have to give it a design. Okay, so whenever you design something, it's basically this model idea. Okay, and so whenever you do this, you're going to be using a tilde. And really, we just care about one group. So we're just going to, one identity of your data set, one piece of metadata, and that is what group is it part of. So we'll just use the tilde and group. If you have multiple parameters, there's really complex ways of signing up this design formula. But most of the time, you're just going to have it like this, okay? And so whatever your column name is, so our column name is group that we care about, you put that in here. And then you have the row ranges here. Okay. And what we just did there, we give it to the DESeq dataset from matrix, and now we have what's called a DDS object. It's convention to call these objects DDS objects. You can call them whatever you want though. But whenever you look for help associated with DESeq, you'll see everyone using DDS because that's what everyone does. Okay, it's just a convention that people use. You can also add, go directly from your range summarized experiment if you want. Okay, so this is one way of generating a DESeq dataset. It's the most from the ground up. You might have a situation where you just have counts and you've got them from someone. And so you can build it like this way. But if you build it in exactly the same way as we did, and you have a range summarized experiment, what you can do is add some extra information to your gene counts. Okay, so you use the col data function on gene counts. And in this case, we're going to add a group information using the $. And this time you just add in that metadata information. Okay, so if you remember, we're just basically adding this to our range summarized experiment. If you do this, you can then just use the function DESeq dataset, and it will build it straight from the range summarized experiment. Okay, so we just use gene counts and we give it the design. And so it'll infer everything else. It will drag it all from the range summarized experiment. So this is nice and simple. Okay, and so now we have a DDS object built in two different ways, and they both work. And so once you have this object, this is your input. And so to do the DESeq operation, there is one overarching function called DESeq. Okay, and so you just apply this to your DDS, and you can assign it straight back to DDS as well. So this is, again, this is a convention. What people do is they'll just keep working with this same DDS object, You don't have to do it if you don't want to. You can name them whatever you want, but this is just a convention, and I'm reinforcing this because when you look for help on the internet, this is what you'll see. Okay, really, this is nice because the DDS object itself actually isn't changing. You're not losing any information. What happens when you perform these steps is extra information is added to your DESeq dataset. So if you compared the before and afterwards, all the original information is there afterwards. It just now has some new information associated with the object. So it makes sense to overwrite it. Oh yeah, so you can see under here, it prints out what it's doing. And so depending on your data set, this can take anywhere from 10 seconds to 10 minutes. Typically, it might be more. I'd say most of the time it's about 30 seconds, okay? But what it's doing is it has these various steps, okay? So it's getting you the scaling factors, looking at dispersions, and then it's making some final estimates before doing a test, okay? And so what you can do is you can look at each of these steps individually and looking at this object. So first, if you want to look at counts, you can use the counts function, and you can provide this argument called normalized to see normalized equals true. And so this allows you to see the normalized counts. And so what the normalized counts are is the counts post-scaling. So DESeq comes up with this normalization factor with which to scale each library, and it's related to the library size. So if you've sequenced really deeply in one library but less deeply in the other, it will scale the really deeply one down so it's more equivalent. And so when you look at compare the counts between one and the other one, they're actually on equal footing. You imagine this is really important because you don't want to think that you just have really high gene expression just because you sequenced it deeper. You can also use the same function to get the raw counts out if you do normalized equals false as well. But so this will give you your normalized counts. You can also have a look at how the variance is being shrunk as well and how that relationship works. And so there's this function called plot dispersts, ests even. So this gives you the dispersion estimates. And this is really one of the strengths of doing a specialist technique like this, the RNAseq specialist packages and functions. So in things like DESeq, we're shrinking the variance, and you can see what the variance was. Okay, in red you can see the fit that it came up with, and then in blue you can see what DESeq adjusts the variance to be. And so this variance shrinkage only affects typically the main body of the genes, and so the ones that are these outliers aren't affected as much. Okay, and so their shrinkage is their dispersion is left kind of high. And so it only affects, well, it affects the majority of genes, but it doesn't affect the ones that are truly outliers or have really high dispersion. But doing this will make it much easier to call significant gene expression, which means it's possible to actually look for significant genes while only sequencing 2 or 3 replicates. If you don't do something like this, it becomes much harder to get differential gene expression. Okay, and so then the final part is you want to actually do get the test out, right? You want to see what is the log fold change between condition A and condition B. And so intuitively, the function you use for this is results. Okay, and so we have the results function here. We could provide it with our DDS. And the key part here is you want to say what contrast do you care about. Okay, I think in this specific example, you only have one real contrast that you'll be interested in, so it's fairly straightforward. But in other cases, you might have multiple different groups, you might have multiple different samples, and so it's really important to specify which one you care about. And so in this case, we care about comparing a cross group, and we want to compare the activated to the naive. Okay, and this order matters. Whenever you do log2 fold changes, so you look at relative changes between two samples, you want the numerator to be the mutant and the denominator to be kind of regular wild type. And so in this case, it's the numerator is these activated cells versus the denominator of the naive cells. And so we do the results function, and we save it as myres, the object myres. Okay. And this is an additional step that's not necessary, but I quite like to do, is I will then order my result by p-value. Okay. So I'm going to extract out my res, the p-value, and then I'm going to use the order function. And what this does, it reorders the data frame based on what is the most significant. And so then you can see here the result, right? You get the gene ID, you have the base mean, the log2 fold change, and things like this, okay? And so what are all these columns? So the base mean is the mean normalized values across all samples, okay? So this gives you a rough idea of what is my base level of expression. Is this a highly expressed gene or a lowly expressed gene? Okay, it doesn't care about the fold change or anything like that. It's just the average. Log2 fold change is the log2 fold change between the two groups. Again, remember, the numerator and the denominator are dictated by how you did the results here. Okay, and you can always see it up here. So actin versus naive. This is in the annotation at the top. Then you have the p-value. This is the significance score, and the adjusted p-value. Okay, and so this means that it's been corrected for multiple testing. Okay, so we have a question from King. Is the order in the results group numerator and denominator? So yes. So it's first the numerator, then the denominator. Okay. So in this case, we'd consider, say, the activated to be mutated or treatment or something like that, and the naive to be wild type or control or something like that. Okay, and so when you have this results object, you can get a quick and easy summary of it using the summary function. Okay, and that will tell you what on average has been the log fold change. Okay, how many genes are up, how many genes are down, how many genes have very low counts, things like that. Okay, I've just noticed it is 12 now, so I'm going to quickly, let me just check. I'm going to cover up to DESeq2 and Salmon now. If you need to take off, that's totally cool. But I'll cover up to that, and then I'll start off with Salmon in the next session, I think. And I think the next section is a little bit shorter, so we should be able to cover everything, hopefully catch up. Okay, so when you're reviewing your data, an important step is also to look at what it looks like, right? You want to plot it. This is often a massive part of the QC, is doing something like an MA plot. Okay, and so what the MA plot is doing is looking at the log fold change versus the mean expression of that gene. And so the plot MA function is quite nice because it highlights the ones that are significant as well for you. And so you'll often see a shape like this, where you have this broad, non-significant pattern at the bottom. And then it tails in, because as you get higher expression, you're able to call differences much more robustly. And so if there's just a small change, it's much easier to call that. And so that's why the fold change ekes in at the end. You can also downweight genes that have a high fold change but low significance using LFC shrinkage. And so what this does is it considers not just the fold change, but it also considers how the significance of the change. And so you can see that this fold change has been shrunk all the way down here, if you compare them, and it gets squished down to the 0 line, because it's essentially worthless data in this place. Okay. And so this shrinkage is kind of nice, because then log2 fold change becomes much more of a useful measure. I typically wouldn't use log2 fold change as any kind of ranking metric unless you've done this shrinkage operation on it. And then we can also convert this DESeq results table into a standard data frame, just using as data frame. Okay, it's a DESeq results object. It contains all this information. It's a nice object to have it in, because you can perform all these operations, but often you want to convert it to a data frame so you can then export it to an Excel spreadsheet or something like that. So you can then do as.dataframe to it, and now it's ready to export with Excel. Yeah, so we're just going to cover multiple testing real quick, and then we'll stop. Okay. So you notice there was a column for adjusted p-value, and hopefully most of you as scientists know all about multiple testing, but just to really drive that point home in the concept of like genomics, this becomes a really big issue. Often we say a p-value of 0.05 is enough, right? And now we have this example from XKCD Comics, where they say there's a link between jelly beans and acne, and they can't find a significance, but then they break it down, maybe it's by color. And they test 20 different colors, and one of them's found to be significant. And so now there's a news article printed out that says green jelly beans are linked to acne. But if you do this process 20 times, that means with a p-value of 0.05, you're expecting a 1 false positive, okay? Because the 0.05 is really just saying there's a 0.05 chance I'm wrong. So if you do that 20 times, then that chance I'm wrong will come up. And so if we're doing this, and now we're not doing it 20 times, but we're doing this over the entire genome with 10,000 genes, that becomes an issue. So there's multiple ways to deal with this. Two of the most well-known are Bonferroni, and the other one is Benjamini-Hochberg, or like FDR. And so with Bonferroni, the way you do this is you take your p-value and you multiply it by the number of genes you've tested, okay? And this is a very rigorous way of dealing with multiple testing. But as you imagine, this can make your p-values very high. It's very, very strict. And so it's not very generous with your data. The Benjamini-Hochberg, on the other hand, does the same, but then it will divide it by the rank of the p-value of your gene. Okay? And so this means it's very, very still strict on kind of your intermediate data set, but on your very top p-values, it's not going to completely kill them. And so that means it's a much nicer way of doing things. And so, yeah, the number of genes you put into the multiple testing process has a large influence on how much this number is affected. So before you do the test, okay, you can't do it afterwards, but before you do the test, you need to consider this. And so some kind of independent filtering can be applied prior to a test, such as removing things with low expression or very high variance. You can't wait until you've found out which genes are differentially expressed and then reapply the correction afterwards to just those genes, because that wouldn't be fair statistics. And that would be biasing your data. But DESeq handles this in a nice way for you because it naturally, when it does multiple testing, it filters out any gene that's lowly expressed. Okay, so this reduces your total gene count down. And what you'll get in your result is an NA in the adjusted p-value column. Okay, and so if you see in this data set, we actually have about half the genes, 10,000, actually have an NA if we use the is.NA, while 13,000 don't have NA. Okay, so it's really cut down on the number of genes that we're actually considering for this, because a lot of the time, a lot of genes just aren't expressed. You can also do this manually yourself using the p.adjust function. And so in this case, we're doing that, and you can see what the value of these scores would be. Okay, and if you played around with your data, you can actually see how much has my data changed because I've done this filtering beforehand. And so you can see the NA values here, and they would all be significant scores of 1 anyway. So it's not like you're losing valuable information at this point. And another key point is typically if you have scores that are NA, you can filter them out for any later evaluation or functional testing. These aren't genes that you really care about because they're lowly expressed and they're not changing. So why care about them? They will weaken any of the significance testing you want to do later on. So if you want to see, am I enriched in a certain class of genes, you can get rid of them and it will speed everything up and it will be a much better test. Okay, so we're going to leave this here. We have there's going to be about 10 slides to pick up on at the beginning of the next session, comparing DESeq2 and Salmon. And then in the next session, we're also going to be covering functional enrichment, okay? And so this really ties into what this late point here is that we're just finishing with, is NAs we can filter out, because in the next step, you want to actually know not just what genes are changing differentially, but what classes of genes. So we're going to pick back up, I think, at 1 o'clock. So sorry I overran a little bit. I talked too much, which is amazing considering it's a 2-hour session anyway. And we will be back soon. Okay. Thank you. And if you have any questions, you know, I'll be here. And switch over to the right share. Okay, cool. I'm just gonna have a quick sip of tea first. Okay, so where are we? What we covered this morning was taking your FASTQ file, checking your FASTQ file looks okay, mapping your FASTQs, so the read to the genome, and then counting it, or doing a direct quantification method with Salmon. Okay. Then we took the counts from the alignment and counting method with summarized overlaps, and we did differential gene expression with DESeq2. Okay, and you remember we took the range summarized experiment, or we took the counts matrix and we put that into DESeq. It does some transformations and it gives us log2 fold changes and adjusted p-values from which we can infer whether a gene has changed its expression between two different conditions. Now, the one missing part of this, right, is how do we take, we can imagine taking those salmon values that we produced, and it looked like a data frame. You can imagine reading it in much the same way as we showed at the end of the first session where we did read.dlim to read in those data files. You can imagine converting that into a matrix and then using DE-seq2, right? There's not too many steps involved. And for those of you who are attending our original sessions, hopefully you, if you don't have those skill sets, you'd know how to look and find those skill sets from reference points to be able to build up that. The nice thing about Salmon is that the people who use Salmon are also R users and also Bioconductor users. So they've come up with ways to go from the Salmon output and put it straight into DE-seq2 to make your life easier. And so to be able to do this, this is just a review of Salmon, which we've done recently, so we'll skip over. There's this package called TX import. So TX transcript import. It's written by the same people that do DE-seq2, so they know DE-seq2 very well and what it wants. And it is designed to take a lot of this different transcript counting software, so not just Salmon, but places like Callisto and other things like that, and make an easy import for those quantified files into a DE-seq2 friendly format. And so I showed you before that we can just do read.dlim and we can have a look. And you can see with read.dlim, we have some information about the transcripts, we have some TPM, we have some counts, right? But this is not the matrix that we need for the input for DE-seq2. And so if we were going to carry on along this way, what we could do is, sorry. Sorry, my ear just popped and now it's making a weird noise. Okay. So when you have your TX import, you need to be able to summarize your transcripts to a gene level and import that into your DE-seq2. And so to do that, this is the main obstacle going from like your Salmon output, which is based on transcripts, to doing DE-seq2 when you mostly care about genes. So to do this, you need a transcript to gene conversion table. And so we can construct this easily from TXDB, right? So if you've built your annotation with TXDB that you used for your Salmon index, then you can just use the same TXDB object and extract out the information that you need. And to do this, you can use the select function. And we've covered the select function in Bioconductor, but it's from the annotation DBI package. There's lots of different selects. There's a select that's in the tidyverse, but this select is from annotation DBI. And what it does is you give it a database of some kind. So in this case, we have our transcript database. You say, what do I want to get from this? In this case, I want to get some information from temp. And so we're saying we want to get temp column 1, which is the name. Okay, we want to get information on the name. And what information do we want? Oh, what is this information? We're giving it a transcript name. Okay, so this is our input for select. We're going column 1. We're giving it transcript names. We tell it this is a transcript name. And what we want to get back is the gene ID and the transcript name, right? And so this is our conversion table that tells us transcripts to genes. That's important for Salmon to be able to be read into DE-seq2. And so the product of this select right, is here. You have transcript name and gene ID. Okay, so these DB objects are really useful for generating conversion tables like this. And so you can get different properties of, say, a transcript. You can get its ID, its exon IDs, its gene IDs, all these kinds of things. And so once you have this table, you can then use the TX-import function to summarize the salmon quantification to a gene level instead of it being at a transcript level. And so all we have to do is provide paths to the salmon files, and we tell it what type it is, okay? And then we provide this transcript to gene document. So you have, if you've downloaded the presentations and all the training materials, in the data directory under Salmon, there is all the quant files for these files. So everything regarding this should work for you as well. And within this, you say, I have this directory, it's data Salmon. I want to look recursively. So this means it's going to look in every single directory within that directory. And it's going to look for any kind of file that's a quant SF file. And you want to get the full name, so it will give you the full directory and the full path to be able to find these files. And so what you'll end up with is a vector which has all the paths to all the quant SF files. So these are your quantifications of the via Salmon. And then now you know where all these files are. There should be 5 of them, right? If you remember, we have 2 for the naive condition and 3 activated T cells. So now you can do TX import on these files. You tell it it's a Salmon type file and that you provide this transcript to gene table that we made. And it reads them all in. And you can see 1, 2, 3, 4, 5 because we have 5 samples. And it calculates the abundance, the counts, and summarizes the length. And so now you have a nice count table. Okay, so if you look at abundance, it gives you a number of TPM values. But if you give it counts, you have the count values. And then there's a separate DESeq function for coming from TX import called DESeq dataset from TX import. And so you can just give it the salmon counts. You can give it the metadata just like we did before, and you can give it and tell it you want to look over group. And then once it's in, you can perform DESeq in much the same way. So you can see here we do the DESeq function for our DDS salmon. We get our results. You can order those results by p-value, and you'll get the same outputs. Okay, so you're just changing the input, and we have this additional import step, which makes importing from a salmon results really easy. And so we've done these 2 methodologies. We've done R subread and we've done Salmon, and I've been continually saying they're broadly the same, or they do very similar jobs. And you can kind of see that here. And so if here we're just looking at the overlaps between the genes that have a high fold change and also are significant. And you can see that the majority are the same. Okay, so we have 170 roughly that are specific to each methodology, but 930 that are overlapping. So there is a large amount of coherence and consistency between the 2 methodologies. That said, you can obviously see there are some differences. So you may find, depending on what type of data you're doing, a preference between the 2. The last thing you can do, really, once you've done this DESeq analysis, and before you might export it into an Excel, is add some extra annotation. So the classic thing that you might do is add symbol, because most people look at the entree IDs and they're just like, what does this mean? Although it's very useful for us because typically these are better quality annotations, this symbol is more human readable. And so again, we can turn back to our select function and we can use the org.db objects to get symbols from it. They also have longer names as well. So if you want a more, even more descriptive name than the symbol, you can get that as well. Whatever annotation you want. And then once you've extracted out your entree IDs and your symbols, right? You have two data frames. One of them has ID and all your results from DESeq. The other one has ID and symbol. So that means you can do a merge, right? Using the ID. And you just give it the information of how you're going to merge it. So in this case, we're doing by.x, by.y. And so this means first column and by rows, okay? And so then when you do this, you don't want all of X, but you want all of Y. Okay, so the reason is all of X would be every single transcript in the genome, but you just want to make sure you get all of your results from your differential gene expression. And then when you have a look, you now have the entree ID and you have the symbol, and then you have your differential gene expression results. Okay, so just had a question, is it common practice to want to do salmon and subread and then merge the differential gene expression? I would say it's relatively common to try out multiple techniques for everything you do, okay? This ensures robustness. What I would say is less common is then doing the merge at the end. If you're happy that there's robustness, then with this, then it's more common to just pick one and carry forward. And then you can also check observations. So what we're going to do now is we're going to check, we're going to cover doing enrichment analysis, functional enrichment. And so when you get to this step, you'd look and say that actually these agree quite a lot. Maybe I'll do the next step with both of them as well and just double check that they both agree, so that when we look at Go terms, they both say similar terms. And then you can say you're really happy with it. And then when it comes to writing your paper, you can focus on one of these methodologies, but in the methods or in the initial writing, the first figure or something, when you're first discussing doing RNA-seq, you can mention, oh, we did it with both these. Normally, this kind of thing gets buried in the supplement, but it just means you have nice, robust results. Okay, so I hope at this point everyone feels like they can go from a FASTQ file to getting some form of counts and then doing differential gene expression. It's not too many steps, and hopefully you can see it's definitely feasible to do yourself on your local machines, though depending on your data set, it may obviously take some processing time. Next set is, I would say, is one of the really fun bits, because you actually get to start to figure out, what does this mean? It's great you get 2,000 genes that have changed expression, okay? But unless you want to go through every single one of them in the Excel and like look at every single gene and see which ones are the top ones and then go, do each of these make sense? That can be quite laborsome. And so you want to be able to get a quick summary of what's going on. And so the most common way of doing this is by looking at enrichment of groups of interest, okay? And so again, we'll be using the same data, and you can get a lot of this information that we talked about. So you can get the count information and the salmon information from here. So we provided a lot of the files that you can use post alignment. So that's all the heavy lifting has already been done. All the counting's already been done. So you can just focus on maybe trying out DESeq2 and getting the results from that, and both from Salmon and from counting. And we also include a CSV file here, which is the activated minus the resting, okay? So this is, or activated minus naive. So basically the results of DESeq2. And so that CSV is a really useful input for what we're going to do now. And so what we're really up to now is evaluating the functionality. So this is immune-related a data set. So really we want to test, do we see immune response? If you don't see any immune response groups coming up, then you have a problem with your data set, right? Because then you don't have any enrichment that you thought you should. And then you can start to look for other interesting things. If I have immune response, are there anything else that sticks out as different or unexpected and worth investigating further? And so to do this, you really need some gene sets. So what is a gene set? Gene set can be anything. It's just a group of genes that for some reason people care about. They've been grouped together because they're in the same function, same pathway, some kind of category that overlaps. And then we can use a method to do some kind of gene set enrichment analysis. And so there's kind of two main ways of thinking about gene set enrichment analysis. There is gene set lowercase e enrichment analysis. And this is considered to be a very broad term for any for correlating sets of genes with a condition, a phenotype, a treatment, whatever you've done in your experiment. And then there's GSEA. So that's with a capital E, gene set enrichment analysis. Okay. And so this was a methodology created by the Broad Institute. And it follows the same lines, but there are fundamental differences in how it works. And both can be very useful depending on the questions you want to ask. And so when you want to do this, you actually need some resources that are generally publicly available. You can, if you want, just come up with your own gene sets. That's super easy and is sometimes a very valid thing to do. But most of the time, you want to be able to compare to these big resources that contain lots and lots of gene sets for you. And so some common ones are things like Go or KEGG or the Reactome or MSigDB. And so if we think about Go, that's the Gene Ontology Consortium. And there's several different categories within Go. It's a very comprehensive list that you can access all of these things online. So if we follow the link here, you can see, you can search for specific things. So if you were interested in autophagy, for example, it will give you all the ontologies associated with autophagy. And you can dig into these and you can figure out which genes are associated with these. And you can really just go to town on this. Broadly, these Go terms are divided into three groups. There's molecular functions. So that means what is the activity of the protein product of this gene? The biological process, what kind of role does that play? So kind of the difference between molecular function and biological process, you imagine, is maybe it's a kinase and that's its molecular function, or maybe in a biological process, it's autophagy related, okay? And so you can see how a gene can have multiple different properties associated with it. And then lastly is, where do you find the protein product? What cellular components is it found in? Is it in the nucleus? Is it in the Golgi apparatus? That kind of thing. And so the way that Go is organized is very much this nested structure, a graphical structure, where the gene sets all the way at the top are really, really big. Okay. And so you can have this term, which is one of the biggest terms, regulation of biological processes. Okay. That's a massive, massive term, and it will include everything almost. And then beneath that, you can have something that's a positive versus a negative. So negative regulators and positive regulators. And so this keeps dividing and dividing until it gets to very, very specific terms. And so you have these umbrellas that encompass multiple goes, and then you have very narrow go terms underneath them. Okay, so that's the kind of the method, like the idea of what a go term is. There's also other things like reactome and keg. And so these are much more focused on metabolism and disease pathways. And so for some people, this is a much more important question if you're really into metabolism and things like that. These data sets are much more specific and focused on those things. And then lastly, of the ones we'll cover, there's also MSigDB. And so this MSigDB is associated with the GSEA and the Broad Institute. And it includes things like Go, but it also includes other curated data sets. And they're divided into these groups you can see here. Okay, and we'll go a little bit more detail into these when we get to that. Okay, but a lot of these things, they have databases associated with them. Okay, so you can see these three here: go.db, keg.db, and reactome.db. And so you can interact with them in much the same way as we have done before with the other DBs. You can do things like select to extract out specific terms. Or you can interact with them by making use of other libraries like GSEA base. And GSEA base is specifically designed for interacting with gene sets. Okay. And so that's where we're really starting. Like with this is we're focusing on the gene sets, and then we'll do the testing in a little bit. Okay. And so the Go terms and everything like that is quite straightforward. But we're gonna take more of a deep dive on MSigDB, because it's an incredibly useful resource and it includes Go terms within it. And so you can get access to GSEA and MSigDB gene set collections on the Broad's website. So if you click on here, you can download the software, and you can also download the data sets. Here, if you click on MSigDB, okay, you can search them, have a look, but you will have to sign in. This sign in is you give them an email, and that's it. Okay, so it's not much of a sign in. It's just them keeping track of who has access, and that's it. So you're not giving too much information away. But within this, they have these different classes of gene sets. They have some gene sets that they call hallmark gene sets. There's positional gene sets, curated gene sets, etc., etc. You've got all of these. And so this is it's a massive collection, and it's generated by scientists. And so it can be from people's studies, or it can be from larger consortiums. But there's a lot of information contained within this. And so when you go on the website and you might find a gene set you care about, or one of these collections, you can download them readily when you follow the links that I showed you. And the download will come in this format known as a GMT, or a gene matrix transposed, which is a GSEA specific format. And the reason they use this is it's a really straightforward tab separated format. You can open in Excel or whatever spreadsheet program you use. And then you can edit it or add extra genes or things like that if you want. And so to interact with this, we can, as I mentioned before, there's this GSEA base package. And this package is really good at interacting with all kinds of gene sets, including GMTs. And so if you were to go to the Broad website and download a gene set, you could just import it into R. using the git GMT function from GSEA base. We also have one of them in the data folder as well, so you can look at it there. And so this is the hallmarks collection, and we can read it in with git GMT, and now we have this gene set collection object. And you can see there's all these different names. It gives you a little summary. We have over 4,000 genes associated with this gene select collection. We have all these different names associated across the top. And you can subset this as a list really easily. Okay, so you can use the single square brackets, and that will extract out another gene set collection, but it will only extract out the first one. Okay, so you can see there was lots of the names here. It remains a gene set collection at this point if you use the single square brackets, but it now just is a length of 1. Or you can extract out just a gene set, so it's not a collection anymore. And it's now, you do that with the double square brackets, much like you can do with lists. If you remember, when you use a list, single square brackets extracts a new list, but of the first entry in that list, double square gets you just what was contained in there. So in this case, we just get the gene set back. And as it's a list, you can also look at what are the names of each of these gene sets. So using the names function. And so you can see that they all start with hallmark, but then you can see what they're related to. So we have TNF alpha signaling, hypoxia, apoptosis, and you can start to infer, like see some of the differences in specificity that we have here, some of the broader terms, but also some of the more narrow terms. Okay. And the other nice thing you can do is you can interact with this with another function called gene IDs. So this extracts out the names of the genes within the gene sets. So in this case, we'll use gene IDs, and this would return a list with every single gene ID for each gene set. And we've specifically subset this to the first 3. But you can see here we have the first gene set, and we have every single gene, which is quite a few. We've got over 100, I think, associated with TNF alpha signaling via NF kappa B. Okay, so one thing that maybe wasn't clear, because I didn't spell it out, MSigDB is a really great resource, but it is focused on human pathology, right? And so if you look at our lists, all of these genes are typically based on human analysis, human experiments, human data sets, which is great. But a lot of us don't work with human data sets. Predominantly, there's going to be a lot of people who work with mice, but then beyond mice as well. And so if you want to work with MSigDB, but you don't work on one of these organisms, you have a couple of, if you want to work with MSigDB, but you don't work with humans, there are a couple of options open to you. So in some cases, there's similar sets. People have tried to recreate MSigDB, but specifically for, say, mouse experiments. Okay, they follow the same ideas and rules that were used to set up MSigDB, but they just switch their focus by switching organism. The other option is use orthologs, right? And so we know what the genes are in humans. Why not just use their relevant ortholog in mouse, and that can be used to generate the gene set. And so both of these are very valid options. The first option, someone has done a lot of this. So there's a project called GSKB. And so this is the mouse genome equivalent of MSigDB. And there is an R package for this, so that's nice. You can use, if you install it, you can then use library GSKB and it'll load it in. And there's 7 collections built into this. Okay, and a lot of them are very similar to what you see in MSigDB. Once it's loaded in, you have to access it using the data function to load that gene set actually into your memory in R. And so here we have data function, and we're specifically interested in the microRNAs. And once you've done that, the microRNAs are in R, ready to use, and then you can check things like names with them. Okay, and they'll be in a similar format to what we saw with the gene set when we read it in with GSEA base. Okay. And so that's GSKB. And so the alternative, right, that I mentioned before is you translate the human to other species using orthologs. Again, someone has done that already. And a lot of that's contained within this package called MSigDBR. Okay, so we can have a look at it. It's a CRAN package, so it's not Bioconductor. So we like it, but we don't love it because we love Bioconductor more than anything else. But there's lots of different species options for this. You can see them listed here. You can get mouse, rat, pig, zebrafish, fly, yeast, and the original human genes. Okay. So there's a lot more options here for you. And so the way that MSigDBR works is once you've loaded it in, you can do library, and then there's a specialist function called MSigDBR. You tell it what species you want, what category, and that will load it all in. The issue with this is it's not in a GMT format anymore, okay? And it's not necessarily a problem, but GMT formats are used by GSEA, and so quite often you'll want to be able to convert it into a GMT format to be able to use it. And so instead, you can see it's in this organization where we have like categories, we have the gene set name. It's a nice flat data frame, so it's really easy to interpret and work with if you're doing anything that doesn't need a GMT file. And so it's fairly trivial to convert it to a gene set though. And so the way to do this is first you need to go through and find each gene set and extract that out. And then you turn that into a gene set, put that into a list, and then turn that into a gene set collection. Okay, so that maybe seems like a lot, but I'll show you how to do it in a step-by-step way. First, you create an empty list. Then you look at the gene set names and you say, how many unique ones do I have? Okay, I want every unique gene set. So we're looking at here. And the way this data frame is organized is it has an entry for every single gene. And so you want every single gene set in your final list. So you don't care necessarily about gene levels. So you create unique gene sets here using this unique function. Then you iterate through every single unique gene set. And you ask the question, which rows contain my gene set? Okay, so we'll start with gene set 1. Which rows contain this gene set? I want to get those genes. I'm going to turn them to a character. Okay, and then I'm going to create a gene set from them. It's going to be the genes in my gene set here, and we'll give it the name, which is the name of the gene set. And then we'll put that in the first slot in the gene list. Okay. And so that means the first slot in the gene list, if we go back to here, will be every single gene ID associated with this first gene set of adipogenesis. Then it will go to the next one, maybe it's autophagy, and it will get all the genes associated with that, and that will go in the next slot. And we'll just recreate the list that way. Each of them will be a gene set, and then once we have this list, it's a list of gene sets. So we can then turn that list of gene sets into a gene set collection. And then once you have a gene set collection, right, you can then do something similar to what we did before. Before we read in a GMT, now we can use the GSEA-based function to GMT to export this. Okay. And so now we've created a mouse hallmarks GMT. Okay, so I've shown you how to interact with GMT files and where to get gene sets in general. Okay, and so these GMT files will be very useful for specifically GSEAs, because that's the file that it likes to work with. But you also, at this point, hopefully can see where we got some of these gene sets and how to maybe, if you wanted to break down these GMTs, they're just basically lists. And I've shown you to go from these data frames to the lists, and you can go back in the other direction as well. And so next, we really need to do the actual experiment, right? We want to see some gene set enrichment. What are my significant genes enriched in? And so, as I mentioned, we're going to approach this in two different ways. The first method is the more traditional way, which is just a simple gene set enrichment. Okay. And so this uses a simple functional enrichment approach. You find what genes do you care about? Say all of my significant genes. These are my interesting genes, and I'll see if they're enriched in anything. The second method doesn't subset your gene list. Instead, it uses a ranking method. And so you'll rank all your genes, for example, by their differential expression. And using this ranking and the ranking score, it can then decide if your analysis or your differences between the groups is associated with a specific gene set. And so we'll see both of these in action. We'll start with the simpler, which is just the functional enrichment. And so at this point, you guys can definitely, everything should be able to code along, because what we're going to be doing is reading in this data file. This is the results from DESeq. And so you can see it contains the symbol, the ID, we've got base mean, fold change, etc., adjusted p-value. And so all these GSA methods, so just lowercase enrichment, all of these typically are a functional enrichment, looking for functional enrichment. And you typically start by filtering your gene sets, or no, filtering your genes of interest down to just the ones that are differentially expressed. Okay. And you can do that. You can also create your genes of interest in tons of other ways. You can look at intersections between different groups. You can look at, as you'll see tomorrow with CHIP, you can look at genes that have a peak next to them, all these kinds of things to generate your genes of interest. But for us, we're doing RNA-seq. So what we care about is what genes have changed, what is special about these genes that change. So there are genes of interest. So first, we're going to filter out all the ones that have an NA value in the adjusted P column. And so this is very easy, because what we can do is just use is.na. We do is.na on the adjusted p-value. And then we can use, if you remember, exclamation mark. This is.na returns a true false, depending on whether it is an NA or not. We want to keep the ones that are not NAs. So we can use the exclamation mark to reverse a true to a false and a false to a true. So that means all the ones that are not NAs will now become true. And then we can subset our data frame with that. Okay, so we put that into the square brackets and there's a comma. So that means we're extracting out all the rows where the p-adjusted value is not an NA. And you can see that here. If you look before, when we looked at the top 3, we had a bunch of NAs, and now these are true values. And we even have something that's very significant here at the top with MDN1. So let's get to the enrichment. A good package to do this is GoSeq. And so with GoSeq, it tests for any functional enrichment within these genes of interest. For an input, what it requires is a named vector of 1s and 0s about whether this gene is upregulated or downregulated, or basically whether it's a gene of interest. Okay, so if it's a 1, it's a gene of interest. If it's a 0, we don't care about it. And so here we're just we're going to focus on genes that are significantly upregulated in this activated versus naive T cells. Okay, you could look at all significant genes if you want, but here we're going to look at significantly upregulated. So to do that, we're going to subset our data set. First, we're going to ask the question, which ones have a p-adjusted value that is less than 0.5 and a log2 fold change that is greater than 0. Okay. So this will give us some trues and false. Okay. We can then turn that to an integer with as.integer. When you do that, any true is turned to a 1, any false is turned to a 0. And then you can add names to this. So we want to add the names using the names function, and we're going to add the entree IDs as the names. And so if you look at what this looks like, you now have this vector and it's named. And it's all 0s and 1s. And if we turn this into a table using the table function, this will count up how many you have of each instance. And so you have 10,000 zeros and 2,500 ones. Okay. So now we want to ask the question, are these ones enriched in anything? And so first we can, with the GoSeq function, you can check what genomes are supported. And so here we use the supported genomes function. We assign this to a vector here, and we just look at the first 2. If you remember, these are MM10 data. So, but this is one of the options, MM10, because it's mouse. And this is where the strength of GoSeq comes in. A lot of functional enrichment packages don't consider this, but there is often length bias associated with significant genes. And so if you're doing analysis like this, looking for functional enrichment, if there's a length bias and say a gene set also has a length bias, then you're going to be enriching, or you have a bias towards enriching that gene set. And so you might find this with very often very core gene processes, right? They have shorter genes, they have less introns, and that's because you just they're trying to get expressed as much as possible. And so things like, say, neuronal genes are often longer and they have more introns. And so this gene length bias can affect what functional enrichment you call. And so the GoSeq actually helps deal with that. And so to do that, you can use the null p function and to assess any of the bias. And so in this, we have our up in act. So that was our named vector we created here. We have the genome. You tell it which genes you want to look at. And so there's lots of different options. But if you were to look on MM10 for the supported genomes, there would be several different names that you could look. And known gene is one of them. We choose known gene because that's what the TXDB is built on. It's the TXDB of known genes. It's a UCSC specific category. And you can see there is this bias in your data. It's not flat. So that's potentially an issue. But GoSeq will handle it when it does its analysis. Okay, so when you do the null p, you save out the output, and that would have corrected the length bias. And then you can now do your GoSeq function, which will actually do the test for you. And so it looks pretty much the same. You do GoSeq. This time we give it PWF instead of up in act. But this is because this is our corrected data. You then also give it what genome builds you care about and the test categories. So this could be a collection, but it needs to be one of these four. Okay, so you can do Go terms for the biological process, molecular function, cellular compartment, or you can do KEGG. And these are all inbuilt into it. And then when you do this, the result is your Go terms that are overrepresented. And so, unsurprisingly, for our immune cells that have had this treatment, we have a immune system response as the top hit. Okay, response to stimulus, defense response. Okay, some of these are very non-specific, but if you dig into the terms, you'll be able to find the more specific ones as you go. You can also search for the Go terms, and you go to here. You can. I should copy that. These terms are universal, so you can then search here to get more information if you wanted to, right? Yeah, we were looking for this. Okay, and so you can follow the link to get detailed information about what's going on. You can also, the Go terms are also, the genes in the Go terms are also in the orgdb object as well. So we can retrieve all the genes in this object using our select again. So we have annotation dbi select. We select this orgdb object. This time we want to get all the Go terms. Or this is our key. This is the keys we're providing as the Go term. Okay. So we're providing a Go term. This is us letting it know, letting the function know we are providing a Go term. This is the Go term, and we want to get back the gene IDs. And so now we have, when we get the response from this, we have the list of every single gene associated with this Go term. Okay, and you do have multiples, and this is because it also lists where the evidence for this Go term comes from as well. So this is a nice, useful thing if you want to focus on very specific types of experimental analysis that derive the Go terms. And so this means we can then filter our differential expression table to only immune response genes if we want to. So you say you find a Go term you really care about, you can then subset your differential gene expression table to just those genes. And to do this, we grab our unique IDs. Remember, there's duplicates, right? So we want to get unique of these ones. And then we ask the question, which of my data frame, so this is my differential gene expression results, which of these are found in this unique set that I have for just immune response genes? Okay. And then it goes in the square brackets because we're indexing and we're just using that to extract out the rows. So this means we'll just get all the rows for the genes that are immune response genes from our differential gene expression table. And so when we look at it, it will look like this. Okay. And so these genes are related to immune response genes. And then we can write that table out as well. So you can save that. And so this is a nice way to start passing down your data into something smaller and more manageable. Okay, so that's functional enrichment, right? It's a very simple test. All it's doing is asking the question, am I seeing more of these genes in your significant genes or your gene of interest than expected? And that's just purely based on how many genes are there, how big is the gene set, how many genes are there in the genome, what is the relative proportion, okay? And then you typically perform something like a Fisher test to test that statistic. It's a very straightforward and intuitive way to ask these questions. The big thing is it requires you to have a gene of interest, right? Having a gene of interest means you have to do a cutoff. Now, it is widely accepted that most people do something like a 0.05 cutoff or a 0.01, but even me saying right there, use 0.05 or 0.01. These are arbitrary cutoffs that we use for statistically derived data. And so we're adding in user intervention at this step. And so it can sometimes be more useful to not do that. And so an alternative method that's really useful specifically for like RNA-seq data is to use something called a GSEA method. And so the way the GSEA works is it looks over your ranked data. So you'll have the genes that change in expression the most, all the way down to the genes that change the most in expression in the other direction. And in the middle, you'll have the zeros, okay? And it will say, are the genes in my gene set enriched in these polar opposites? Are they most enriched right at the top, or are they most enriched right at the bottom? And it does this by doing what's called a random walk. And so as you start at the top of your gene, your most significant genes, or your genes that change the most, this would be all the way over here as a left on your data set, okay? Okay, and actually, yeah, you can see here how they're ranked, right? So you have gene set A, gene set B, they're high over here and low over here. So that means the fold change between here and here is strongest at the top, but then it completely switches by the bottom. And so you can then look at the gene set on top of this. They're marked in this as a bar. And you can see there's quite a lot clustered at the top. And say maybe you cut your significance like here or something. And if you cut your significance here, say this is 0.05, actually, like there's a lot of genes up here, but maybe it's not more than you'd expect compared to the rest of the genome. But if you're looking over the whole thing, there does seem to be a tendency for more at the top. And so that's why the GSEA might be useful. And so if you now switch this 90 degrees and you walk in kind of this direction from left to right, what happens is every time you every time you hit a gene in the gene set you're testing against, it moves in this direction away from the 0 point. And every time it takes a step that is not a gene set, or not in the gene set, it comes back to the middle line. Okay, so it's starting at gene 1, and it's saying, is this in the gene set that we're testing against? If it's a yes, it goes away from the 0, and if it's a no, it moves back to the 0. Okay, and it keeps doing this. And so if you imagine you have an enrichment of genes in this kind of polar end, you have this enrichment and lots of genes in this gene set, you're going to move further and further away from the 0, because it's going to take a step away from 0 every single time it encounters one of these genes. And so it will do this, and you generate a plot like this. It then will do a permutation where it compares what you see versus if you just randomly distributed which genes were significant and not, and you repeated this 1,000 times. Okay? And so this is a permutation test where it compares against this permutation to ask, is my random walk looking really different to if it was just essentially background? And so if you see a big swing out here or a pattern like this often means that there's a significant deviation from what you'd expect. Okay, and so we have a good question here. How is the leading edge defined? And so a leading edge is a term used in GSEA as these are the genes that are the ones driving the gene set enrichment. Okay, and so the leading edge is defined by anything that is to the more extreme end of the peak of enrichment. Okay, and so what I mean by that is when you hit the peak in your random walk, so you can see here, this is what is used to generate your enrichment score. Anything to the left point of this is considered to be in the leading edge because it's driving this. Anything after this, you have less genes than maybe expected at this point. And so these ones aren't considered to be interesting. So it's a statistically defined method to be able to, without you having a cutoff, find the genes that are the most interesting. That's the nice thing about it. Okay, and then Nathaniel asking another question, I guess a more basic question, how do we know when to reverse the effect on the graph? +1 first, then -1 afterwards. Yeah, so if it's going in the other direction, so once it gets to the midpoint or the 0 point, and say you're looking at these genes that are going from low to high, if there's an enrichment in that, you'd expect it might go up a little bit for a bit, and then it will come down below this. So this is only showing the case of if you have enrichment in like a positive upregulation. But if you're actually checking like this negative upregulation or there's negative gene sets, that's also picked up. It will just be the graph is coming down here instead. But it takes care of this for you. Okay. And so the big thing about this is you need to generate a rank table to go into it. We saw how to make a GMT earlier, and that's your gene set that's going to be going in, but you also need your ranks. And so the rank format that goes in, you would typically use either the stat column from your DE-seq result or log2 fold change, but only after you've done shrinkage, okay? And so the reason is the stat column or the shrunk LS log fold change that's been shrunk has information about the amplitude of the change, but also the significance. And so these are important metrics to be able to use in your GSEA, because these are the genes you really care about the most. And so you can extract these values out, and you can extract out the entree IDs as well. So we have the IDs and we have the stat here. We're using stat. And then you can just write it out as a table. Okay, so it's just a 2-column table. So we have 2 columns and we do write.table. Okay, and you should have this available to you in the data if you want to look at what the product is. It needs to be tab separated. And so this rank file you can then use with GSEA. Okay, so once you have the GMT and the rank file, GSEA has an online tool that you could use if you want. But there is a package to use it within R as well, which is what we always prefer. And it's called FGSEA. So that stands for fast GSEA because it's quite quick. And so we can grab our GMT pathway. This is the one we generated earlier, the mouse pathways. We can have a look at it. And then we have our rank file that we just saw. We can read this in if we want. If we're going to use it with FGSEA, we actually have to turn it into a named vector, just like we did for GoSeq. Okay, but then instead of ones and zeros, it's going to be our values. Okay, so our stat values. And so to do that, we can extract out the first column. And that will be the stat value. And then we can assign names from the row names. Okay, it's pretty straightforward. Data frame manipulation into a vector. And then we run FGSEA. And so there's several things we can add as options for FGSEA. We're going to, obviously, we give it our gene set and we give it our genes of interest or our ranked gene list even. You can control what gene sets it actually looks at. So maybe you don't care about gene sets that are actually only a couple big. There's quite a few gene sets you'll find that maybe they only have 5 or 6 genes in them. And maybe that's not of interest to you. So you can put a limit on this. You can also put a max size. So some of the really big gene sets, like just regulation of biological processes, that's almost all the genes, and it is not informative at all. So you can cap this. And then the number of permutations. So if you remember when I was explaining GSEA, right, it does the walk. It looks at what your data looks like. And then it does it 1,000 times, but with just randomized data. And then it sees how yours compares to those distributions and sees if yours looks like an outlier. It doesn't look like what would happen if you just did it by randomly. If 1,000 is a good number for this, but if you're like short of time, you can cut it down, but obviously you're reducing the statistical power. You can also boost it, but again, that would add time. Okay, and so once you do this, you get your result, and the result is saved in here. Again, what we often do is we'd order the result by the most interesting value. So in this case, we're going to order it by NES. Okay, and what NES stands for is the normalized enrichment score. And then we'll look at the first two rows. Okay, so we have a pathway. So that is the gene set, basically. So interferon gamma came out of this. We have the p-value and the adjusted p-value. So we're still doing multiple testing. So that's good. We have the enrichment score and the normalized enrichment score. So these are basically how equivalent to a fold change, how much enrichment do we see, and gives you an idea of how much enrichment you see in this specific group. You can get an idea of also, this one's, I guess, is the most important one, is the leading edge. Okay, and so the leading edge is all of the genes that are basically considered to be significantly part of this group. Okay, the size is the size of the group, and the N more extreme is how many, when you did the random permutations, how many of the random permutations actually had a more extreme distribution than your observed. And so if you have 0, that means your observation is the most extreme, which is good because that means it's probably the most significant. Okay, and so you can extract out the things like the leading edge. And then you can do it similar to what we did before, right? You can then subset your original data set based on the leading edge and output that if you want. I just realized the time. Sorry, I'm overrunning again. I'm almost done. So if you are fed up of GSEA, we're pretty much there. But I do have a nice little extra thing, which I recommend people trying out just at the end. Okay, so to actually visualize the plot that I kind of showed in the demo while I was explaining GSEA, there is a function called plot enrichment. And what the plot enrichment function does is you tell it which thing you care about. So basically which gene sets, and you provide it your result. And then it will plot you a nice GSEA plot. Okay, and so you can see this here. This is definitely enriched, right? You see every single tick here is one of the gene set members. And so you can see they're definitely all the way up at the left. And this is a very steep curve. Okay, so it's definitely enriched on this left-hand side. Okay, so I've just got like 3 or 4 slides at the end that won't take very long, just to introduce a final tool that we have started using a lot here at the BRC. It's called Cluster Profiler. And the reason we like this tool is it does everything we've covered so far, but it does it in one place, and it generally has more uniformity, and that makes it much easier to work with. And it has some really nice plots. That said, it doesn't have quite the same customization options or control over the parameters. But I imagine for you guys who are just getting started, this might be an easier tool to run with. We just haven't had time to exhaustively explain it. And so the way the cluster profiler works is it allows you to look at things like Go, KEGG, MSigDB, custom gene sets. And it has a very, very good, what's it called? Textbook associated with it here. And it explains everything that it does. And it takes you through all the way to generating plots that look like this, and including networks and stuff like that. Okay, and so we're actually, Doug, who's taking the ChipSeq course tomorrow, is going to cover more of a nice example of this tomorrow, but I just wanted to show you quickly how easy it is. And so in this case, maybe we want to look at C7 of MSigDB, because this is our immunological signature. And so Cluster Profiler just needs a 2-column data frame with the gene set names and the gene IDs. And so we can extract that from MSigDBR really easily because it's the 3rd and 4th column of this data frame that gets output by MSigDBR. And then we need to grab our gene information. So again, I'm just going to grab anything that is significant from our GGE data frame. And then I just put it in this function called enricher. Okay, so I grabbed all my significant genes. I get the first column, which is all their entree IDs. I have my term to gene table, which I got from here, the MSigDBR. And I run an enricher on it. Okay, and so this does the functional enrichment similar to what we did with GoSeq. And then if you want to do GSEA, it's pretty much the same function. It's just called GSEA instead, but you give it the same kind of named vector as we did for GSEA. You can just give it this same term to gene, and it will run a GSEA on it. And then all the subsequent functions will work in the same way as the functional enrichment for making plots and things like that. And under the hood, it's still actually using FGSEA. So it could be an easier way to do exactly the same as what we did before. And so then inside is this special object called an enrich result object. Inside is lots of information. Okay. You can see stuff about all these different IDs that get identified. But you can also turn this is a non-flat object. You can also turn it into a data frame, just doing as.dataframe, and then you can export that. And then it has a lot of these plotting. So I showed you very briefly in the textbook that it has online, all these different plotting options. One of my favorites is just dot plot. You can control how many categories it shows, and it will just show you what are these top hits, how do they overlap with these gene sets. And so in this case, you can see that a lot of these are related to CD8 T cells, okay? And they're specifically related to these experiments. So you can see here, naive cells, okay? Which would make sense because we've gone from naive to active T cells. So this is good. And then the last thing that is really nice about these is these plots are also all built in ggplot. So for those who attended the ggplot course, you can then modify them in exactly the same way with ggplot. So here I'm playing around with the text size of the y-axis because these are really, if I left them the length they were or the size they were, because they're so long, they would take up the entire ggplot frame. Okay, so that is gene enrichment. We have caught up to where we need to be. Again, sorry for overrunning. The next and final section, we're going to talk about different transcript analysis. So this is looking at isoforms. And we'll also touch on some ideas about how to work with multiple groups. We will start that at 3, I believe. Yeah, at 3. And as always, we have these exercises here. So you can we have examples in here where you can start to play around with GoSeq. We run through this data set that is different to the one that I'm doing in the main course. It's a liver versus kidney data set. We have all the resources for it in our data file. So you'll just be able to load them in and start to play around with them and see what's going on here. So you can start to try and generate plots that look like this. Okay, it's actually a really nice set of exercises. So it's a really good way to supplement what I've been talking about. Okay, so I'll be back in 45 minutes. If anyone has any questions in the meantime, chuck them on the chat. And thanks for sticking with this. I know GSCA is maybe a little bit complicated and a lot to get your head around, but once it makes sense, it can be a very useful way of interpreting your data. Okay, right, well, bye for now. Okay, so chat around so I can see chat. There we go. So all ready for the final part. So what we're going to cover in this final session is differential exon usage and how to visualize that as well, and some nice plots we can make. And then finally, when we get to the end, we'll just touch on some of the ideas that we can put together when you have multiple different groups. So say you're comparing 3 different tissues together. How do you start to kind of put the blueprints together for that analysis? So we have a quick question from Adam about the recordings. So the recordings are not up and available yet. I'm working on this at the moment. We plan to put it somewhere public for everyone to be able to access. I'm very busy with the actual course content at the moment, just making sure the day-to-day is going okay. But it will happen, and it will happen soon. Okay, this is actually the last session I will be directly leading, so I will have a bit more time to do that. And I will make sure everyone who's signed up knows exactly where they are. So don't worry, you won't miss out. Okay. Actually, just because there's a couple new names, one thing also to mention is if anyone needs help with any of this outside of training time, you can always reach out to us. And so just to exemplify that, there's the Getting Help button here. If you click this, this will take you to our dedicated GitHub, and you can raise an issue here. So you just click New Issue, and well, I'm not signed in on this browser, but you can click New Issue, and you can give us a question. So this morning we got a question from King while he was setting up this, setting up for Salmon this morning, which we covered. And so I was able to give him some feedback on how to do that and instructions. And what we do is we try and leave up some of these messages so that if someone has the same issue, they can come back, they'll see it, and they'll get the answer without even having to write an issue in the first place. But likewise, we have similar things here, and we figured out how to get it working. Okay, so we try and keep a record of all the things that we're doing to try and help you guys, and you can always come here for help. And it's a nice, easy link straight from the course. Okay. Likewise, we have all these nice exercises here with the solutions. And so we can help with any part of this. You just got to give us a shout. Okay. So we're going to go to the slides. Okay. So most of the time in the sessions that we've covered so far, we've been covering this data set where we have these activated T-regulated cells, T-reg cells even. And so that was a really nice data set for differential gene expression. However, to make it easy for you guys to see what's going on for differential transcript analysis, we're using a different data set, and this is a PTBP1 RNAi. And so for those of you not in the know, PTBP1 is a really important factor for splicing. And so this is a nice example data set to use for this, because a lot of things change when you manipulate or do RNAi against this factor. So that's why we're switching data sets, because we'll have some really nice positive data to look at. And so prior to this data has undergone mapping from FastQ to BAM using R subread, and it's been counted in both genes and exons using summarized overlaps. Okay, and so you remember when we did this earlier, we were talking about using disjoint exons, and so that enabled you to map to specific chunks of the exon which are unique to different transcripts. And so you can find the data that's already been processed in this directory. So if you've downloaded the data for today's training, if you go to our course and then the data directory, you can just get this, in essence, our data object. And so to get it into your session, you'll be doing a load. And so what we're going to be trying to do is now really identify different exon usage to then infer what transcripts are changing. And so this is how the data was generated, right? It's the same as what we did earlier today. We have a file list containing BAMs, a BAM file list. We have, in this case, 3 replicates. One, these I believe are the controls, the vector, right? And this is the RNAi targeting PTBP1. And we're going to do maybe a band file list, and we're doing yield size to limit the amount loaded into memory. And so to be able to do this, we're going to be using a package called DexSeq. And so DexSeq is a package that's available in Bioconductor that is designed for differential exon usage, hence DexSeq. And you may notice, obviously, it has a very similar name to DEseq, okay? And so the people who came up with DexSeq really try to use DEseq as a blueprint through which to generate the workflows for DexSeq. And so that means once you feel familiar with DEseq, using DexSeq uses the same workflow, same kind of rules, similar outputs, and that just makes it easier to follow the rationale. Okay. And so one of the things we need for DexSeq is we need to have the annotation. And so if you remember, we use the disjoint exons function. And this is our way to generate a annotation that has all the different exons that have been disjointed. Okay. There is a Python script in the DexSeq package called DexSeq prepare annotation that will take a GFF file, and do this with the GFF file. And so if you have external data, external annotations, this potentially could be the easier way. But you could also, with external data, you can also load it in as a TXDB object and still do disjoint exons on it. So it's up to you. It depends on the level of annotation you have and whichever is the way is most comfortable. But this is just to highlight that this is available to you. And when you look at the DexSeq packages notation and vignette, it will talk about some of these things. And so that's what's there. But just as we did this morning, you can use the genomic features package and TXDB object for humans. So because we're doing a human data set, and the disjoint exons function from genomic features can be used on the TXDB object to get your disjoint exons. Okay, and then we add these custom names, okay, which has the gene ID and a number associated with the exonic part. Okay, so each exonic part in a gene has a number. And so each part of the annotation then has a unique ID, which you can use, which is its name. And so this is what it looks like. We have some exons here. And we have their IDs here as the names. We have the gene ID, transcript name, and the exonic part. Okay, and so this is pretty much what we just created, right? And so you can see the complexity really in this section and this section here. We have a bunch of different transcripts associated with this gene, HSH2D. And you want each of these exons to represent a unique pattern of these transcripts. And so maybe the easiest to see is this one is this middle section is only found in a couple here. And there's some skinny sections that are found in only a certain isoforms here. Okay. Meanwhile, we have over here, we have a large chunk that's found in a bunch. Well, this one is only found in a couple of them as well. I'm finding it hard to differentiate between them. But basically, it needs to have a unique pattern of presence within each transcript. Okay, so if it's present in a couple of them, that's fine. But it will cause a cut every time there's something unique. This is a much better example, actually. Okay, so you can see here that this first chunk is present in all the transcripts. This next chunk is present in some of the transcripts, so this one and this one. This chunk here is present in these two transcripts, but it's also present in this transcript, so it actually becomes a separate chunk. Well, this final chunk is also present in all the transcripts. Okay, and so it's basically your list of options of where these exons are found. And this gives DexSeq all the information it needs, right? And because it can use that, the counts in each of these regions to then infer which transcripts they belong to. So first, we need to do some counting. We do counts over this disjoint exons, okay, using summarize overlaps. So as we mentioned, this data is all available to you at this point. And so we now have counts at every single exon. Okay, and so this, we're using the assay function to access the range summarize experiment. And we're just looking at the first couple of rows. And so across in the columns, we have the different samples, and the rows, we have the different exons. So we're just looking at the first two disjoint exons. And as always, it's a range summarized experiment, so we can use the range row ranges accessor to get out the actual exon IDs back. Okay, but yeah. Okay, so now you've got your counts. This is what we've generated. You've got your counts. It's been done on disjoint exons. Now you actually want to investigate what is significantly changing. So what we were doing previously, right, is we'd look at gene A or gene A in sample 1 versus sample 2, or condition 1 versus condition 2. And so here we have 2 reps, and we'd look at the difference, and we would go, oh, there's lots of expression here, and there's none here. So there's a significant change. But here, if you actually compare this one versus this one, if you did DE-seq, it would probably think it's about the same. There's not actually that much change in the global amount of gene expression. But you can actually see there's a clear difference in the transcript being used. So although there's still this shorter transcript is being used, you can also see evidence for the longer transcript also being used. So you can see that here and here. So we're making 2 transcripts from this gene. And so this is why we create disjointed exons, right? Because you're not sure which transcript the counts have been assigned to. And so you can use information that's specific to, say, just one transcript or a combination of transcripts to infer which ones are changing and which ones aren't. And so this is what goes into DexSeq. And so the DexSeq is the option we're going to be talking through today. As always, there's other options. Voom and Limmer provided a good method for this as well. DexSeq can be a little slow, but there are ways to parallelize it. And it has some really nice outputs. It's been put together in a very considerate way. Voom and Limmer, however, is very fast, but once you have the result, it's much less useful to work with. And so we generally prefer, it's like worth the compute time to get the DexSeq running. And so for our inputs for DexSeq, it is very much similar to DE-seq too. So first we have to create our metadata column. And so this is the same. So we'll have a condition or a group or whatever you want. And so in this case, we have our RNAi treatment. And it has to be a data frame. And it's a data frame with row names. And the row names correspond to the sample names. The column names correspond to the groups that you care about. And whatever's in that column says which group it belongs to. Okay. And so this is super easy to generate. And so previously we used a function called DESeq dataset from matrix for DESeq2. There is a similar function called DECseq dataset, which can be used in the same way. And so you have to use accounts matrix just like before. And then we have to provide maps for the exon IDs and gene IDs. So you can associate which exons go with which gene ID. So we can generate our account matrix just using the assay function. We can get our G ranges using the row ranges function. We can get this all from our ranged summarized experiment. And then we want to get the gene IDs and the exon IDs. Okay, and so we can get the gene IDs from our G ranges. And the exon IDs, we can actually just use the row names from our count matrix. Okay, so at this point, we have a ton of information, and it's pretty much everything we need to get this to work. Okay. Now, okay. When we do this analysis, we're actually interested in a much more complicated level of differentiation between our groups than when we did it previously with DESeq. This is because we're actually interested in differentiating changes at an exon level between samples. And so when we created the design formula before, what we did was we just did a tilde and then we did group of interest. But now we do this much more complicated design formula. Okay. And so this, if you want to get into the weeds with how these design formulas work, you can do some research really and look at these different design formulas. And there's a lot of different more nuanced ones you can use with DESeq and even DECseq to ask different questions. But most of the time, we're just asking the same thing. And so you can just use this one here because this is the one basically that you'd most of the time care about. And so the only thing you'll have to change is change group of interest to whatever name you have in this column. Okay. And so in our case, this is going to be changed to condition. And so then this is us running DECseq here. So we load in DECseq as always with library. We provide the count matrix. We provide the metadata. This is our design. We've updated it to include condition. We tell it what the exon IDs are and what the gene IDs are. Okay, so now it can use the count information to aggregate up to gene level information. And of course, as with DESeq2, there is a simpler way of doing this that goes straight from a range summarized experiment. Okay, so there's a DECseq data set from SE. Okay, and so that means you can just add again in much the same way. You can add the metadata information directly to your range summarized experiment. So in this case, we're adding to we do col data function, and we add specifically a column called condition, and we add a factor, which is our different group informations. And then when you do that, you don't have to specify things like exons and genes, because that's all saved in the ranges. You don't have to specify which groups they're in because that's specified in the coal data. The only thing you need to give is the design information. So this is a nice little shortcut to get around it. Okay, so at this point, you've got your two methods for running with DexSeq. This first way is the more complicated and more explained way, where you know all of the inputs going in. And you can imagine, again, if you have external counts that you haven't worked through step by step, maybe you've got them from somewhere else, you can input it like this, right? This gives you a way to get something in that maybe wasn't in a range summarized experiment. But if you've been following our workflow, you can do the nice straightforward way by using the DexSeq data from SE. An additional step we recommend you do is you filter out exons or filter out genes with low exon counts, or exons with low counts even, not genes. So as you can imagine, DexSeq is a much more complicated algorithm, right, than DESeq. It's not just applying a simple statistical transformation and test. It has to understand what each transcript is, and then infer based on the levels at each transcript or at each exon, which transcript is changing. And those exons might be present in 3 out of 4, and then the other one might be present in 1 out of 4. And so it has to combine all these proportional information about how often that exon is included, and compare them to each other to be able to then say, actually, it's this transcript that's changing versus another one. And so that, yeah, it's very, very time consuming. And so removing data where possible is going to be beneficial. And so to do this, what we do is we just take out any exons that aren't expressed very much. And so a good rule is remove anything that don't have at least 10 counts in them in at least 2 of your samples. Okay, but you can look at your data, see what looks like, you can draw some curves, see what looks like at the low end and looks like it's not going to really generate anything. And if you want to change away from 10, you can start to get some intuition of this as you start to play with your data. Okay, and so the way we've done this is we use an apply function. We use assay to grab the counts. So now we have a count matrix. We're going to use apply. So it's going to apply to assay this function, and it's going to do it on a row by row basis. And it's just going to be summing up how many are greater than 10. Okay. And if that number is greater than or equal to 2, then you should be good. Okay. And then this generates a true false for to filter. And you can then just filter down your data set based on that true false. And so it's just filtering out the rows based on that. And so you can see when we do this, we actually filter out 110,000 exons, 108,000 exons out of, and 160,000 are kept. Okay, so we've trimmed out 1/3 to 1/2 of our data in this way. And so this can really help you later on with the computation. Okay, and so there's a step-by-step way of doing this, just like with DESeq. You can, there's several functions to estimate the size factors, estimate dispersions, and then do the final test for differential exon usage. And so you can do these individually. And you notice each time, instead of creating a DDS object, it creates a DDX object. And again, this is just a name. It doesn't necessarily matter, but it's just convention. So when you look at the handbook and places like that for additional help, you'll see this a lot. And what's happening is this: every time you run one of these functions, same with DESeq, it's not removing anything, it's not changing anything, it's just adding more information into this object. And so every time you run it, here, you're not losing anything as you go. So there's no point keeping each object. So you might as well just overwrite it each time. And so then by the time we get to the end, we actually have estimates of how the exons are changing. And so doing it in a step-by-step method might be useful for you if you want to break down each of these things it's doing, especially if you're doing it locally and it might be a bit much for your computer. Maybe you're using your computer at the same time. So like you can do step 1, do some other stuff, then you can do step 2. And take it one piece at a time. You can also just do the DEXeq results function though, and it will do everything in one go. And so what this does is it produces a results object, and you can order this by p-value, just like we did for some of these other objects in the past. So we can see at the top what are the most significant ones. So yeah, that's exactly the same thing. Oh, no, sorry. Sorry. So there's all these steps, and once you do these steps, you can use the DEXeq results function to get the result. This is similar to DESeq and the results function, okay? And this, you'll end up with your result at the end. This is the function that will do everything for you, okay? And it includes the results section of it as well. So this will go all the way from a DDX object, which you created at the beginning, all the way through to your results object. Okay, so this single piece of code here, this single function is equivalent to these 4 and this one. But again, like I said, you might find it better with your computer to do it step by step, or you might find it easier to understand what it's doing, especially to begin with. Okay, so we create this result object, and we probably want to have a look at what it looks like. So like I said, it's been ordered by p-value. And this is the first row, okay? It contains a lot more information than DESeq2, so I'm only going to show you one row of it. But what we have is some group information, feature ID. So this is like gene and exon. What is the average expression of this exon? So this is the base mean. If you remember, that's just the average across all your conditions. We have information on dispersion, the test statistic, p-value, adjusted p-value. So it's 0, so that means it's very significant. And then we have also, what are the count values in senescence and SHPTB? Okay. What's the log2 fold change? And then we have some genomic data about where it is. We have the transcripts it's in. We actually have the count data here. So this has this, I believe these, where is it? These 2 here show the average for each case. But in this case, it shows the counts for every single individual one. And we can turn this into a data frame. Okay, so this is the same result, but we can just turn it into a data frame. And so you've done all that. Again, tables are cool, but really we want to be able to show this, put it in at some kind of presentation so we can show everyone what cool transcript usage changes we've actually been able to show. And it has some really nice inbuilt functionality. So in this case, we're going to do plot DexSeq. Is this? Yeah. So this is our top gene, 57142, right? That's its gene. And so we can then have a look at see what's happening with this gene. And it produces a plot that looks like this. And so on the left is its expression level. And then on the bottom is the exons. And the exons are ordered from left to right by the ones that are changing the most, I think, or most significant. And then you have these arrows which point to the exon, so you know where they came from. And so you can see the most significantly changing ones are exon 8 and 9, and also exon 5. Although this one is changing very minimally. You look here. But exon 8 is changing from something like 50,000 down to 1,000. So that's a 50-fold change. And that is this large exon here. You can also add the argument to display transcripts. And so this is the same result, but now it's showing every single transcript available. And so you can see, yeah, it's this one that's changing. So we can infer there's been a change from one of these top transcripts to one of these. And as it's changing to both of these, it's probably this one or this one, right? And actually, as this one isn't changing, it's probably going to be this one right here. So we have this one being made. And so what we do is we then follow that up. We can look at IGV, and you can see that this exon here is no longer really there, and the little one next to it as well have disappeared. So we've gone in one case, we're basically going from, I think, this or this isoform, yeah, this isoform to this isoform here. And so you can do this with any of them. They can get a lot more complicated, obviously. That was quite a simple example, but here we have a lot of exons that are changing. But it highlights all the ones that you should care about in pink. And then you can look at it and you can see. So it's mostly these two, but there's also slight changes here and some in the middle and up the front. So these two are the main ones. And yeah, there's potentially some changes here, but there's also, yeah, I think you can see that quite clearly, actually. It seems there's maybe more expression here in the bottom one, but overall the green is expressed less. So there must be a lot more increased inclusion in this final exon versus the red one, while the opposite is the case for these two exons here. So you can see quite a lot of difference. And then you can visualize these DexSeq results in a HTML report. So hopefully, I've tried to fix this link this morning, but I haven't actually tested it. No, it doesn't work. Okay. Okay, so this link should be in the data directory. So I'm just going to find it real quick and show it to you. It is called test 4. Okay. Okay, so let me just switch over to this. And so this is a report created of this specific gene. and you can look at it, and it's much bigger and maybe easier to interpret. But you can look through, there's lots of different information all contained within the report. So you can look at the counts. So there's obviously variation in the counts here between the two. But if you look at transcripts, there's also significant change in the transcripts, specifically to this one. Okay. So this is a nice little report that you can make. I need to find my way back. There we go. And it's super easy to do. And you just need to give it the gene that you care about. And so it's nice being able to make these HTML reports like this, because if you do have collaborators or non-bioinformatic people that you work with, such as PIs, right? If you want to really drive home what is all the data relating to this isoform change, easy, create this report and they'll have everything at their fingertips and they can really start to explore it themselves and get an intuition about what's changing. And so from what we saw, we were creating a p-value and an adjusted p-value for every single exon, right? But you might want to get something that's much more akin to a value for the entire gene. So you can say this gene is changing as opposed to this exon is changing. And that can help you narrow things down quicker. And so one way to do that is there a function called per gene q value. And so you do this on your results object. And so this is a step we often do. We'll do per gene q value, and we'll get a list of, well, it's a nice numeric vector. I'll come to your question in a sec, Simon. And so you get from per gene q value using this function on the results object. You'll then get a numeric, and then you can take this numeric and add it back to your original data frame. So this is a step we often do. So our data frame, which contained a per exon level change in exon usage, right? That has the gene information in it. And so this also has the gene information in it here at the top as the names for this vector. So we can use the merge function to merge them together once we turn it into a data frame. And then when we do this and look at our data frame again, you'll see it's right at the front, but we now have the gene Q associated here. And the adjusted p-value is here as well. So you can see, actually, this is kind of interesting, that at a gene level, yeah, actually, it's not that interesting. Both these exons are very significant, and it manifests at a gene level as being very significant as well. Super significant leads to super significant gene level changes as well. That's not surprising. But you can then have confidence that this a change at the gene level and not just at the exon level. And so to go back to Simon's question, how does it dictate the order? So in these images that we have included in the slides, it is dictating by significance level, I believe. So the most drastic changes are on the left. But I'm curious whether we can control this. So I'm just going to quickly change my sharing. Let's go to help. I should load ATACseq in first. And then we'll just do question mark plot ATACseq. Okay, so you can see there's lots of options. Let's see, display, transcripts, names, legends, color. So you can change the colors. I could not see if there's any control here, but you were right. It did seem on the in the HTML report, it's done in it's all the order of the exons is maintained to be a representative of the gene, right? Which I think potentially makes more sense. If you then also have a nice table that's showing you which exon is most significant. So I can show you that. Yeah. Right. So the ordering is kept. So I prefer it this way. But when you do it in R, and it's like this, the ordering isn't kept like that. I'll have a little dig. I'm assuming there's a way to control this because there is a difference between how it's output in the HTML and how it's being visualized when it goes straight in through R. So one of the parameters must control it. But when I just had a quick look in R in the help page, there wasn't anything that jumped out as an obvious answer to that. But yeah, sorry, I can't give you a specific way to solve this, but this is actually where Google might come in as a helpful friend, because someone would have asked this question before. Okay. So we were, yeah. Okay, so that's differential expression and differential exon usage, and how to see which transcripts are changing. Either, yeah, we've covered either how to see if a transcript is changing because there's a gene level change in expression, or if a transcript is changing because it's a different isoform. And the last thing we'll touch upon is starting to work with multiple groups. Okay, and so as I've alluded to, this is just some ideas about some loose ideas about how to start to manage this working with multiple groups. There are a lot of very useful tools to take this a lot further, and which we'll try and expand this section at some point in the near future expand this section of the training. But for now, this is just to give you a little primer to get you going. So far, we've only been comparing two groups, right? We compared that Treg cell between active and naive. But it's fairly straightforward to compare lots of tissues or multiple samples. It's something we routinely do in the BRC commonly. And so in this case, we have some tissue data set, which you can access here in the data folder. It's tissuefull.rdata. And this is actually the data set that's been used most of the time in the exercises. So it contains, I believe, kidney, liver, and heart data. And so you can see here, we load this in. And we have 6 columns. We have sorted heart 1 and 2, sorted heart, liver 1 and 2, and there's 2 more in the middle that you can't see because it's abbreviating everything. But they're kidney, okay? So just like I said. And so what we can do is when we're generating our range summarized experiment for DE-seq, we can have all of this in it together at once. There's no point separating out for all this. There's rarely a point. Sometimes there is, and that's getting a little bit into the nuances of how DE-seq works. But most of the time, you can just put everything all together. And so what we're doing here is we're adding the metadata into the col data using the col data function in range summarize experiment. And we're going to add it to a section called tissue. And so tissue is what we're going to be caring about. So when we do our DE-seq dataset directly from the range summarize experiment, we'll say the design is over tissue. We can then run DE-seq, and we can just use the results function, but this time we just change what we're comparing. So we can compare heart versus liver, we can compare heart versus kidney, and we can have both these sets of results. And so we're just using the same tool set we already know, how to do pairwise comparisons, but we're just running it multiple times. And we just do it in the same object because that makes life easier for us. And so it's as simple as finding the genes that are upregulated in one, and then finding the genes that are upregulated in the other one, and doing a comparison between the two. Do I have a lot of overlap? That's a really good starting point to start to understand what are the differences between tissues. And so before you can do that, you have to remove NAs. Okay, so what we're doing is we first convert our results table to a data frame. We then look at the adjusted p-values and we remove anything that's in NA. Okay, so if you remember, is NA asks, is it in NA? You get true or false. You flip that on its head when you add the exclamation mark at the front. So false becomes true, true becomes false. And then you keep anything as a result that isn't an NA. And then we're going to subset this down to just the log2 fold change and the adjusted p-value, because that's all we care about. And so now we want to stick everything together. So we want to make sure we have some good column names so we can understand what the product is from these column names. And so we're going to do paste heart versus kidneys on the existing column name heads. So the column named heads were log2 fold change and adjusted p-value. And now we're doing, for the heart versus kidney object, we're just adding the fact that we know that this is the heart versus kidney object into the column names. Okay, by pasting that onto the existing column names and then reassigning it back. And so now once we have these two data objects and we know what the columns are separately, we can now put them together and we'll still know what all the data is. And so we can use the merge function. And so we use the merge function and we give it both a data frame, so it's going to stick them together. And the by we do for it is 0. So that means when you put a 0, it's going to combine them based on the row names. And so the row names in this case are the gene IDs. So it's going to look down, figure out which genes match, and then stick the log2 fold changes for the heart versus kidney with the log2 fold changes for heart versus liver. And so now you have one data frame that contains all your data for the comparison. And so you have your adjusted p-values and fold changes for heart versus kidney and liver. And so now it's all in one place. You can easily start to extract which ones are significant in both, which ones are significant and up in both, or down in both. So we have this object called full table, and we're going to ask, which one fold change in heart versus liver is over 0, and the fold change in heart versus kidney is over 0. And the p-adjusted values in both cases are less than 0.05. And these are considered to be up in heart. And then we can subset our data by that. And if you look at that, the result, we can look at the first two columns and you can see we have these fold changes, big fold changes in both. And it's nice because they're kind of scaling with each other. So that's good. And the p-adjusted values are scaling with each other. So this would suggest that whatever these changes are, they're very consistent and they're proportional in the tissue. So it means it's robust across tissues. And so you can imagine making a logical data frame about whether each gene was upregulated or downregulated in both tissues. Okay. So in this case, we're just going to ask it, is it up? We're going to create a data frame. We're going to ask the question, is it up and less than 0.5? And that's going to go to this column up first liver. And we'll do the same, but for the kidney. And then we can look at this data frame we created, and it has a nice false, true, false, true. And we can just give that straight to make a Venn diagram to see what's going on. And so Lima has a nice inbuilt Venn diagram function. There's lots of other, there's a package also called Venn diagram. And I would say a lot of the time Venn diagrams are not the best way to show data, but in this cases like this could be a nice way to show what's going on. And so you can see that there's a significant amount of, well, maybe not significant statistically, but there's a lot of overlap between what's happening with the kidney and the liver. And you can also see there's a lot happening that's specific as well. So using approaches like this, you can quickly subset your data using pretty much all the skills you know already to be able to parse out which ones are the interesting genes for up and down, or only up in one or up in both. Really depends on your question. And so the other way that you can really tackle this, or one of the other ways, is using something more akin to, instead of a pairwise test, something more like an ANOVA, where you're saying, you're not asking the question, is my genes expression different between liver versus kidney? It's more that I have liver, kidney, and heart. Does the gene expression fluctuate across these samples between these groups? And so to do that, we use something called a likelihood ratio test, or an LRT test. Okay. And so that means we're going to use a more complicated model here. And so when we supply a model of 1, that means there is it's a no groups model. And so we set the parameter of reduced to the alternative model, okay, of no groups. We also set the test to LRT, so that's our likelihood ratio test. And we just run DAseq just like normal. Okay, so you can use the DDS object you've already created. At this point, you're doing a different kind of test, so I'd export it to something else. So in this case, we're doing DDS2. And then when you run results, you can just run results on it, okay? And then you'll get a results table that looks very similar. And so when it does this, it's really just assessing, is there information in the tissue grouping, essentially? Can we see information in this grouping and compared to no grouping whatsoever? And this is how the ANOVA test works, and you'll get an adjusted p-value as a result. And so you'll get a bunch of genes that are significant that are changing across this. So this is just a different way to be able to test within your multiple groups, is something changing? And so this, for me, we're not going to cover this now, but this is a really nice way of starting to subset your data to just genes that are interesting. Often I will want to start to draw heat maps of genes that are changing, or I want to do clustering or something like that. This is often the start point for that when you want to go into clustering, especially when you have many groups. And so we're not going to cover the clustering, and we're not going to cover like making these heat maps. but often we'll start at this point and then we'll go into the visualization later. And so once you've done your LRT test, you can actually look at some of the ones that are significant. Oh, how does it generate a log2 fold change? Yeah, so the log2 fold change isn't that useful after an LRT test. If serves me correctly, my memory serves me correctly, it is comparing everything versus the first group, I think. I think it's that one. But essentially, it's not a useful metric at this point. The useful metric is the adjusted p-value, and that's pretty much it. But you can check, the manual is really, one thing I haven't said, DESeq 2 and the creator and maintainer Mike Love is some of the most responsive and exhaustive people when it comes to managing their package and supporting the community. So the manual on using DESeq 2 is very long, but also very easy to understand. It has, DESeq 2 obviously has some very hardcore statistics underneath it, but he does an incredibly good job of explaining it. So if you want to get into the nitty gritty of DESeq 2 or really start to use it for lots of things, the manual is a great place. And then if you start to break it or you can't get it to work in your situation, he is also very responsive on, say, the Bioconductor forum and places like that. So that's a really good, he's a fantastic resource for the community. Okay, so once you have your result after your LRT, you can actually use things like plot counts to start to subset to some of these interesting genes. So we have this one here that's come out and it's interesting, 1788. And so you can just plot counts and it will show you the counts across all three tissue types. And so you can, as with what we did before, we can start to combine things like our pairwise t-tests, we combine them together, and we combine that also with our result from our LRT, and we just put it in one big data frame. And then you can export it into Excel or whatever you want to do with it at the end. But it means you'll have all of the results of your data in one place, which is nice. So in this case, we're just doing a bunch of merges to merge together our paired results and also our all groups results. Okay. And so that's actually one thing I just wanted to touch on. Like, this is a really nice way of visualizing what's happening, patterns across several groups. But I think what I touched upon, this is often a starting point for me for multiple groups analysis. And so what I will do is I will subset this to just the ones that are significantly changing. And then I'll put it into there's a package called pHeatmap, and I'll put it into that. And then I can see what's happening to I put the counts into that. And so then I can see what's happening to my gene expression across all these groups, all these samples, but only looking at the ones that are actually changing. And so that's a really nice way of doing things. Or I can then put it into some clustering to get k-means or hierarchical clustering. If you want to get patterns like up, down, up versus kidney, liver, heart, or something like that. But that's, yeah, this is just the kind of the platform to jump off into that. And so if you have questions about that more in-depth stuff, you can always meet with us, and we're always willing to sit down and talk about analysis with people and give people help. But yeah, so this is going to be us for the day. There is some exercises here. A lot of it's to do with, we have some comparing some data sets and looking for KEGG enrichment using GoSeq. Okay. And then a little bit about doing some differential transcript usage. So yeah, this is some good exercises to practice what we've covered, but obviously it's been a long day, so I don't expect you to jump on that right now. Hopefully I'll see some of you tomorrow. I'll be, as always, in the background lurking, but Doug will be taking the session tomorrow on ChIP-seq. It will cover similar workflows to what we talked about today, but how to apply them to a very ChIP-seq specific way and how to QC chip. And yeah, so thank you very much. I will stick around also in the background if people have questions, as always. Yeah, well, thank you. Yeah, thank you, everyone. Have a good rest of your afternoon.